Hello, my name is Bruce Williams. I am the president of the C.L. Davis Foundation and a senior veterinary pathologist at the Armed Forces Institute of Pathology's Department of Veterinary Pathology. Today I would like to take a couple of hours, or a number of hours, to talk to you about a area that a lot of pathologists are very uncomfortable with, and that is ultrastructural description. I thought it would be a good day to wear a gray shirt, since almost all the pictures we'll look at today are black and white. Before I start the talk, I would like to thank a couple of veterinary pathologists who have provided a tremendous amount of material and inspiration for this lecture, and that would be Dr. David Fritz of the United States Army Medical Research of Infectious Disease, and Dr. Dana Scott, one of my residents, and who recently finished his career with the Army and now is working at the Rocky Mountain Laboratories in Montana. With that, let's go on to the first slide. Okay, I'm not really sure why pathologists have such a fear of ultrastructure. Perhaps it's because we tend to think in pink and blue, and all of these images are shot in black and white. Perhaps it is due to a bad memory of high school biology, where we learned many of the techniques and many of the organelles that we will talk about today. Actually, I find ultrastructure one of the easiest pursuits for a pathologist once you have the ability to wrap your head around what you're expected to do. We're going to focus on the use of descriptive techniques in ultrastructure. This is something that very few pathologists get a chance to do, as EM scopes are being shut down all around the country. Every couple of years there's a new technology that people believe will replace ultrastructure, and I found that that is not exactly the case. Many years ago, when flow cytometry first came out, people thought that this would replace electron microscopy. Then it was in situ hybridization, or fluorescent in situ hybridization. This will replace ultrastructure. Immunohistochemistry is widely used and has replaced a number of times, at least in our laboratory, where we might have previously sent a piece of tissue to be looked at under electron microscopy. But we've found that there are still a number of times where we require electron microscopy to get to a definitive diagnosis. For example, here at AFIP we have a large department of nephropathology, which looks at glomerular changes, especially those that are seen with glomerulonephritis. And still today, almost every case, probably every case that comes into that department is evaluated by electron microscopy. And we're going to talk about that later on. When we talk about the morphologic description of ultrastructure, I know a lot of great ultrastructural pathologists who really have trouble putting what they see down on the written page. Most of you who will be looking at this video tutorial will be preparing for a certification examination, and you want to know just what you need to know to get through that question or several questions on your examination. I'm going to try and focus on that. I find that if you do your morphologic descriptions on ultrastructure the same way over and over, it is very easy to get into a good habit. Morphologic descriptions for ultrastructure are easier to write than they are for your standard H&E slide, because there's a very particular way that you want to do that. When I look at a morphologic description, one of the most important things is to identify where you are. And most electron micrographs only have 2 or 3 cells to look at. So it's very important from the first to state the tissue or cells that you are looking at. If you're confident you're looking at lung, first thing that I would put in your morphologic description would be lung, period, and then go on to the rest. If you're looking at brain, we'll start with brain, etc. Okay, so the first word in a good ultrastructural description is generally the tissue that you're looking at. Now, ultrastructural descriptions generally have 3 parts. You can put them down as independent paragraphs, or you can just put one paragraph with 3 distinct parts. The first thing that you need to include in your morphologic description of electron micrograph are the normal findings that you see. This is important because it goes back to support the tissue or cell that you listed as your first word or sentence. Okay, so if you believe that you're looking at liver, this will be all the normal findings that are associated with hepatocytes, polygonal cells, tight junctions, centrally located nucleus, abundant glycogen, numerous mitochondria with short, stubby cristae. All of the things that you can identify in those cells which support your diagnosis of a particular tissue should be in that first paragraph. And we're going to take a look at all of this as we get farther into the lecture. The second thing that you would put would be abnormal findings, things that deviate from normal in terms of evidence of degeneration. The vast majority of ultrastructural or electron micrographs that you will be asked to describe will have fairly distinct evidence of degeneration. Now, generally, necrosis is not readable on electron microscopy. When you have a necrotic cell, all the proteins congeal, they clump. What you have is a black mass of osmophilic material. It will be totally black. It's very difficult to pick out cellular detail. So when we talk about change within the normal cell, it's usually degenerative and has not proceeded to actual frank necrosis. Finally, the last part of your description will be things that have been added or lost, usually in terms of infectious agents, which could be viral inclusions, they could be bacteria, abnormal compounds, certain mineral compounds or elemental compounds like lead, for example, leave very characteristic inclusions. So we have 3 distinct paragraphs: normal, abnormal, and abnormal findings or agents. Okay, this is the way that I was taught 20 years ago by Dr. Dave Fritz to describe electron micrographs. And this is the way I teach my residents today. It's worked well for 20 years. It is a very easy way to get a good flow in the description, and I will be emphasizing this over the next 5 or 6 hours. In terms of your EM vocabulary, it is a fairly specialized vocabulary, but believe it or not, you know many of these words. Is many of the same words that we use when we look at H&E slides. For the purposes of an examination, you really don't have to be an EM expert. And obviously, this is not something that I can teach you in 5 or 6 hours. What I would like you to do is develop a vocabulary that will allow you to sound like one. I'm going to try and teach you some very basic concepts about electron microscopy and how to correlate that with what you know about light microscopy. In terms of trouble words, when I'm looking at descriptions that students have made for electron micrographs, there are certain words that jump out at me that let me know that the person describing the slide really is unsure or doesn't know what they're looking at. For example, the term structure or organism are very non-specific terms that are big red flags on your papers. And we'll go over some more of these. The overuse of the word vacuole. There are certain times in which the term vacuole is appropriate, and that is generally when you cannot identify the organelle that it's derived from. But a lysosome is a lysosome, not a vacuole. Endoplasmic reticulum is endoplasmic reticulum, not a vacuole. So certain words like this let me know that someone is in trouble in their description. One of the things that people who don't do this on a regular basis will incorporate into their description is the term electron dense and electron lucent material. And in terms of grading these papers, you're never going to get points for use of these terms. It sounds good, but it means nothing. And just like when we describe glass slides, okay, the purpose of description is not only to describe something, but also interpret what it is. If your descriptions are replete with, there is abundant electron-dense material, chances are you're not going to do very well on the exercise. The diagnosis, the bottom line diagnosis in ultrastructure is very similar to what we do with glass slides. A 1 or 2 or possibly a 3-sentence morphologic diagnosis. And ultrastructural diagnoses tend to be very simple. Okay, you will obviously always incorporate the organ and the tissue that you are looking at. Remember, when you are looking at only a few cells, the name of the tissue is often not sufficient. In the liver, you may be looking at hepatocytes, or all the cell types that you may be looking at are biliary epithelium. So it's important to make it very clear what tissue we're in and what section of the tissue you are in as well. Here we're using the term liver, hepatocytes, to be perfectly appropriate. As we said before, the vast majority of morphologic descriptions will be simple degeneration. It is uncommon to see an inflammatory lesion, an itis. It can be done if there are a number of obvious inflammatory cells within your micrograph, such as neutrophils, lymphocytes, macrophages. An itis, like a mastitis or an encephalitis, may be appropriate. But in most cases, electron micrographs usually focus on the characteristic cell in an organ and show various stages of degeneration. If you find that you are using the term necrosis in your description, as we said before, you are likely in testing examination not looking at it appropriate. So we don't see a lot of inflammatory lesions when you have 3 or 4 cells. If you are looking at a neoplasm, and we do use electron microscopy today to diagnose a number of neoplasms, such as amelanotic melanomas, which don't stain with our typical immunohistochemistry or histochemical stains, then that morphologic diagnosis simply becomes 2 words, such as laryngeal rhabdomyoma, cutaneous melanoma. Okay, you don't need to worry about anything more than that. What I would like to do today is to break this lecture up into a series of components. The first thing that I would like to talk about is very general and applies to almost every cell type. We're going to look at the important vocabulary words, the important organelles and structures that you need to know to craft a substantive electron microscopic description. We're going to extract a lot of information from what we know about light microscopy. And as we all know, there are 3 basic types of cells. We have epithelial cells, we have mesenchymal cells, and we have round cells. And each of these cells have very specific structures that are contained within them, that keep them together, and to keep the body together as a whole. I want you to take a look at this cartoon. We'll start with epithelial cells in our discussion, and this is a prototypical epithelial cell. It could be from the respiratory tract, it could be from the GI tract. And I'd like to point out a couple of things that are common to all epithelial cells. If we look at the top of the cell, epithelial cells, generally, all of them have a microvillar border. We'll look at this very carefully. Okay, quite a few of your epithelial cells, especially the gut, are absorptive in nature. and the presence of microvilli allow a lot of absorptive area, maybe 10 times what we would see if the cell was flat on top. So when you see microvillar borders or a microvillus, you're definitely dealing with an epithelial cell. Now, absorption, in the most part, is a one-way process. You want your epithelial cells to absorb from the luminal or the microvillar border, to pass this material through the body of the cell so it can be processed, and eventually to be excreted through the basement membrane into the vascular system. What you don't want is things to scoot down in between cells. So epithelial cells are joined together to each other by a number of specific structures. Okay, we have tight junctions, and tight junctions we'll look at very carefully in a minute. Okay, they're a little different. They are tight. They generally are up at the top of the cell. They prevent material from coming down in between cells. They are the first line of defense. But they also have areas which allow materials to pass between cells. We tend to think of a cell as a very self-contained entity which is concerned only what's going on within itself. But remember that cells are connected. They pass chemicals between each other to communicate. One thing I want to do within this lecture is to get past some of the preconceived notions that you might have about cell biology from when you learned it in the 9th grade. Okay, so moving on. Near the top, at the base of the tight junction, there is an impermeable junction that goes around the periphery of the entire cell. And these are known as belt desmosomes. And nothing really gets by. You can't absorb anything. Everything, if it gets through the tight junction, it stops right here. And desmosomes are structures that are primarily composed of filaments. In epithelial cells, they're composed of keratin tonofilaments, which are very strong. And this gives the desmosome its strength, and it's very difficult to pull these cells apart where they are connected by the belt desmosome. There are a couple of other structures between cells, binding cells together or connecting cells, which are also important. We do have the belt desmosome at the top of the epithelial cells. Scattered along the rest of the cell-to-cell border, we will have the occasional spot desmosome. They're not always in the same place, but the network of spot desmosomes gives additional strength to cell binding, especially in areas of increased tension designed to pull cells apart. And finally, we have these gap junctions. The gap junctions are places where cells communicate. There are a number of chemicals that are passed between cells, whether they are neurotransmitters, whether they're chalones which turn on or turn off growth, whether they are substances that increase absorption, decrease absorption. Cells communicate with the cells around them. And gap junctions are one way that you can pass transmitters from one cell to another. We said that desmosomes bind cells to each other. Well, at the bottom of each epithelial cell, there is a basement membrane, and this one is represented by 3 distinct layers: the lamina densa on either side and the lamina lucida. And that's a generalization about basal lamina. They don't always have 3 layers. Some have more, some have less, but every epithelial cell sits on a basement membrane. And they don't just sit, they are fixed on that basement membrane by hemidesmosomes. Hemidesmosomes join epithelial cells to the basement membrane so they don't come off. There are certain diseases, like pemphigus, which attack proteins in the hemidesmosome, such as desmoglein, weaken them, and then the epithelial cell surface will split right at the basement membrane. So this is a very prototypical epithelial cell. Let's look at some of these in more detail. At higher magnification, you can see here are your microvillar border. Okay, directly under that is the tight junction, which is composed of the tight junction itself and an intermediate junction. Okay, and notice that there is a little hole here, and we'll look at it in just a minute at higher magnification. But this is a place where materials can pass from cell to cell. Here is a spot desmosome. These are densities of microfilaments or keratin tonofilaments with which epithelial cells are replete. And they all come straight into this desmosome and reinforce it. And we're looking at the condensation of all of these microfilaments coming in from various directions, and they condense right here at the desmosome. Another way that a lot of cells bind to each other are these filamentous projections, just little fingers which give the cell additional surface area for binding. So these are connecting structures. These are present only in epithelial cells. If you see microvilli, if you see desmosomes or hemidesmosomes, the chance is that you're looking at an epithelial cell. Once again, higher magnification, where we can see this tight junction, we can see the intermediate junction or connexon, where these intercellular communication chemicals are passed. And here I want you to see these desmosomes, these spot desmosomes, and you can actually see all of those really tough keratin intermediate filaments going into that desmosome and lending it structure and strength. And I said before that desmosomes were characteristic of epithelial cells, and they are to a certain extent. However, you can see keratin filaments forming desmosomes in some mesenchymal cells as well. We are looking at macrophages from a minnow which are surrounding bacteria. And you can see that this macrophage, which is a mesenchymal cell, is forming a desmosome. It's not something that we expect, but in lower vertebrates, in fish and in amphibians, there are certain cells which are capable of forming desmosomes. Let's start on some of these organelles. And as I said before, I'm not going to make you an expert ultrastructural pathologist today. That's not the intent. But the intent is to give you confidence in analyzing and describing electron micrographs and show you that for the most part, you already know the vocabulary, you know the different types of organelles that you need to craft an appropriate description. If we take a look at this structure right here at the top, this is a nucleus. And here we have a nucleolus. The nucleus obviously contains the vast majority of the DNA within the cell. DNA is not only the genetic material, but it is where proteins are translated, where they are transcribed, okay? And then this RNA will go out into the cell and attach to the machinery that actually makes the protein. These are the blueprints for all the proteins of your body live here. What we can see ultrastructurally is that there are large areas in which the protein or the nuclear material is lucent, and there are areas where the nuclear material is clumped. Okay, the appropriate term for this lighter material is euchromatin. These are protein strands that are unfurled. These are This is where the active transcription of protein occurs. Around the outside, generally around the outside of most cells, is clumped dark heterochromatin. Heterochromatin is essentially junk DNA. It's either bad code, okay, all the bad stuff is junk, it's not used, or it is DNA that encodes for rarely used protein. But the stuff that isn't used generally accumulates around the edge. And you can tell a little bit about a cell by the amount of heterochromatin versus the amount of euchromatin. If the nucleus contains only a little bit of heterochromatin, then that cell is considered very mitotically active. It is transcribing and translating a lot of proteins. If you have a lot of heterochromatin, as we see here, it is not translating a lot of protein. At the bottom of the image, I want you to look at this structure right here. This is known as the plasma membrane, and it's not a simple single lipid membrane. It actually has a potential space. It is a bilayer, and it communicates with the endoplasmic reticulum. And one of the concepts that I want you to realize is that this cell is incredibly active. And at any given time, there are millions, perhaps billions of proteins which are being created, translated, taken from their blueprints in the nucleus. And messenger RNA has to go through from the nucleus into the cytoplasm where all the ribosomes are located, where the protein's actually made. And one of the structures that it passes through are these nuclear pores. And we're looking at the wall of the nuclear membrane cut in cross section, right here, just cut tangentially. And what you can see are these little round holes. And this is where all the messenger RNA comes out or comes back in, because sometimes messengers from the cell surface cause changes in the cells that make the proteins shift, protein creation shift in the nucleus. So all of these little bits of messenger RNA come in and out through these nuclear pores. About the only time you'll ever see that. Sometimes people, when they get a fortuitous cut through the nuclear membrane, will still see these structures and will talk about them as viral particles, and they couldn't be any further from the truth. Often near the nuclear membrane, you will see condensed, irregularly round structures which are known as nucleoli. Nucleoli are also centers of protein production. I'm not going to talk a lot about nucleoli, because in terms of ultrastructure analysis and description, they're not all that important. Certainly you would like to note the presence of nucleoli. A metabolically active cell will have very large nucleoli, and they often have multiple nucleoli. Think about cells within tumors which are highly malignant, very aggressive, and reproduce at a high rate. Those nucleoli will be very large. They will be prominent because that cell is trying to turn itself over and over again. And it requires a tremendous amount of protein production. Nucleoli often have two parts. There are more condensed areas known as filamentous sections, and there are more lucent areas known as granular sections. And the euchromatin projects into the granular section, giving them a more lucent appearance. That's about all I'm going to say about nucleoli. They're there or they're not. They're often by the nuclear membrane. And they are one of the earliest organelles to disappear, even before the chromatin is lysed. Those nucleoli will disappear in early stages of cellular degeneration. This is an organelle that we all know, and we've known it since we learned about cells in middle school. And this is the mitochondrion. The mitochondrion is the energy factory of the cell. And believe it or not, you can tell a tremendous amount about a cell when you look at the types of mitochondria, the construct of the mitochondria, and the location of the mitochondria, as we will see in the future. The mitochondria are surrounded by a lipid bilayer, a membrane, which projects into the inside of the mitochondria and creates these stacks of cristae. Okay, the cristae is where oxidative phosphorylation takes place, the creation of ATP. And as we will see shortly, nothing happens in a cell without ATP, except for perhaps diffusion. So without ATP, cells die extremely rapidly, and changes occur very rapidly. In between these cristae is the matrix. And the matrix of the mitochondria is where anaerobic glycolysis is made. The cell, if the cristae is not working, can still make ATP, but it makes it at a very lower level, much more slowly, and less efficiency, so you will have the byproduct of heat given off. Now, when we look at mitochondria, what I want you to do is I want you, every time you see a mitochondria, to take note of the length of the cristae and what proportion of the mitochondria they make up. One of the concepts I want you to understand is that cells that have a high metabolic requirement for energy, for example, muscle, will have many cristae. They will be very long, they will be very prominent. Cells that do not have a high metabolic requirement for energy will generally have mitochondria which have short, stubby cristae, and not many of them. And it tells you a little bit. Eventually you'll become very familiar with the cells that have high energy requirements, those who have low energy requirement. And this is a bit of corroborative evidence that will help you to prove that the cell you're looking at is muscle, or is liver, is brain, is a neuron, is a biliary epithelial cell, etc. Scattered throughout the matrix are these little dark aggregates of mineral, generally calcium, and mitochondria as they age tend to accumulate a little bit of calcium. However, in degenerative changes, they accrue a tremendous amount of calcium, and they will eventually hit a critical point where the amount of calcium within the mitochondria will poison it, and it will no longer be able to make ATP. Look at these mitochondria, and look how thick the cristae is, how many they are, and when you cut them on cross-section, they have these vesicle-like appearance. These are known as tubular vesicular cristae, and they are very common of certain types of cells. The cells we see, tubulovesicular cristae, are cells that are producing steroids. So there would be cells of the adrenal cortex, cells of the corpus luteum, or the interstitial cells of the testis are all excellent examples of cells that have tubulovesicular cristae. Whenever I see something like this in an electron micrograph, I immediately think of adrenal cortex. But it could be, there are a number of cells. Any cells that make steroids. There are even cells within the kidney, cells within the lining of the stomach in certain species that look very much like this because they are producing prostaglandins. And we know that those areas, the vasculature is very sensitive to hypoxia and requires a constant influx of prostaglandins, PGE and PGI, to maintain patency. And if you look at those cells, you will see mitochondria with these tubulovesicular cristae. Let's leave the mitochondria and go to the cytoplasmic protein factories. And we are looking at cross-sections of endoplasmic reticulum, which are studded with these small black ribosomes. And this is referred to as rough endoplasmic reticulum. Now I want you to take a look at this particular section. You can see that the walls of this endoplasmic reticulum are lined by numerous ribosomes, which makes a rough endoplasmic reticulum. But there are also numerous ribosomes which are free in between the cristae, floating in the cytoplasm. Those are also ribosomes. Okay, you can have a polyribosome unit if you have an aggregate of ribosomes, which makes similar proteins, but they can also be single within the cytoplasm. And the importance of this, another important concept, is that a ribosome, their location may change from hour to hour or day to day, and they may receive instructions to join the rough endoplasmic reticulum, or they may receive instructions from the cell to pop off and be free in the cytoplasm. As a general rule, ribosomes that are lining the endoplasmic reticulum, or the ribosomes of the rough endoplasmic reticulum, are creating proteins which will be exported from the cell. Those that are free in the cytoplasm will create proteins that will be used within the cell, whether they're going to be used as part of the nuclear membrane, whether they're going to be used in the mitochondria or in any of the organelles as structural components. I can't tell you. Okay, but now let's take that concept one step further. Cells that have tremendous amounts of rough endoplasmic reticulum are cells that will secrete protein from the cell, such as plasma cells, which make antibodies, which is secreted from the cell, or cells within endocrine organs, or cells that make something like osteoid or cartilage. So osteoblasts and chondroblasts have large amounts of rough endoplasmic reticulum. Cells that generally don't export a lot of protein. All cells export some, and all cells create protein to continue the life of the cell. But cells that do not export protein will have large concentrations of ribosomes free in the nucleus. We're going to take a look at that later. Another very important concept I want you to know is that ribosomes are the source of protein production. They're made of protein themselves. They're almost entirely protein. And as such, areas of the cell that are high in protein will be blue. Okay? They have an affinity for hematoxylin. And we'll look at this more closely later on as we start to go through all the cells and what makes a particular cell a particular cell. But any cell that has a lot of rough endoplasmic reticulum or a lot of free ribosomes, the area of the cytoplasm where that is concentrated will be a dark blue color. There's another type of endoplasmic reticulum. And we are looking at the liver of a dog, and I want to start putting some of these concepts together. We're looking at the liver of a dog, which has a tremendous amount of endoplasmic reticulum. But if you look at it, it does not have ribosomes studding it. It is smooth endoplasmic reticulum. Smooth endoplasmic reticulum serves a number of purposes. It is, and the reticulum, the endoplasmic reticulum communicates with that plasma membrane. Remember, a lot of your protein is being transcribed right there. It's being created, and that's occurring in the endoplasmic reticulum. So it is much easier for messenger RNA to emerge from the nucleus into the plasmalemma and go straight into that endoplasmic reticulum. A couple more notes about smooth endoplasmic reticulum. It is also the site of detoxification in the body. It is a site of cytochrome P450 oxidase production, and it's used to detoxify compounds. Certain cells, such as the liver, as we see here, have large amounts of smooth endoplasmic reticulum because they are primary sites for detoxification of injurious substances. We're looking here at a dog who has been on anticonvulsant medication for a while. And anticonvulsant medication is a low-level toxin, which we give to dogs because we prefer that we can't see that the liver is being intoxicated, but we can't see the dog is having seizures. So when we put animals on anticonvulsants, we walk a very fine line between poisoning the dog irreversibly and solving the seizure problem. Now look at all the smooth endoplasmic reticulum. You can almost not see anything else here. And this is because over time, as we give this very low dose of a poison, the body will adjust and it will realize the need for increased amounts of cytochrome P450 or cytochrome oxidases, and it will cause a tremendous proliferation of smooth endoplasmic reticulum to handle that drug. And that's what we see here. The change takes place over hours to days to weeks. It is considered to be a chronic change in the lifespan of the cell. As we said before, we're going to start putting some of these concepts together. Something else I want you to look at very carefully in this cell is the mitochondria. Take a look. They're very small. And look at the cristae. The cristae are short. They're very stubby. And this is what I call, this is not a real term, but I like to call them couch potato mitochondria. Not because they look like potatoes, but obviously this cell does not have a high metabolic demand. And liver cells generally don't because they live in a low oxygen tension environment. They don't have a lot of oxygen to contribute to the creation of ATP. Liver cells, especially those in the center of the lobule, basically live at the edge of hypoxia for their entire lifespan. That's why when we have shock, when we have sludging of blood, the cells of the central lobule of the hepatocyte are the first to go. So these are couch potato mitochondria, and this is a feature of hepatocytes. They do not have a lot of oxygen, hence they have adapted to a low oxygen environment. Another structure that you will see that is extremely important, not only in the production of protein, but the correct addressing of protein, is the Golgi body. We're looking at the Golgi body. The nucleus, if we could see it, would be in the upper right-hand corner. This is a curved set of stacks or vesicles. And what happens is the protein will come, or the messenger RNA will come from the nucleus, and it will hit the first membrane, where within this Golgi complex, that protein will be addressed. It's like the post office of the cell, and it tells that protein whether it's going to the cell membrane, for example, to be part of the lipid bilayer, whether it will go to become part of a ribosome. It will actually, the Golgi tells the proteins where they go in the cell. Additionally, it goes past each stack, and a different protein will be added to tell it to move to the next stack, the next stack, the next stack. And by the time it gets to the convex surface or the trans surface, it will be completely addressed. It will know exactly where it has to go in the cell, and off it goes. In terms of utility of the Golgi apparatus for diagnosing cell types, it doesn't have all that much utility. Cells that create a tremendous amount of protein, such as a plasma cell, will have a large Golgi, which we can see which we can see under light microscopy as a pale area very close to the nucleus, which we call the HOF, a light microscopic term. I can also tell you that the Golgi apparatus appears to be extremely hardy. It is one of the last organelles to breakdown following the loss of ATP in a cell or during cellular degeneration. As I said before, we have a tendency when we're unsure to use terms like cystic structures or in ultrastructure, vacuoles. Now I'm going to show you a couple of structures here that are very important, and I want you to take pains not to call them vacuoles, because when we can tell what a structure is, we shouldn't be calling it a vacuole. One of the organelles that's very common in especially inflammatory cells, but all cells have them to a certain extent, are lysosomes. Okay, we have a number of lysosomes that are extremely important. You have phagosomes when the body engulfs something, whether it's a bacteria, whether it is a obsolete or effete part of the membrane, it encloses it in a phagosome. And here are phagosomes right here. These are a special type of phagosomes, okay? They're called multivesicular bodies. And when you see a structure like this, okay, it is filled with little bits of the cell membrane that are worn out. And the cells recycle. They try and conserve useful bits and pieces of themselves that they can reconvert, they can break down and use again. Okay, now I can't tell you exactly what this particular multivesicular body contains. Could be cell membrane from one of the organelles, which have addresins no longer needed. Could be the nuclear membrane, it could be the cellular membrane. But this multivesicular body is filled with little bits of membrane, which are going to go back, and the body's going to try and conserve as much as it can. Now these phagosomes will combine with a membrane-bound structure known as a primary lysosome. A primary lysosome is present within the cytoplasm. It is waiting to fuse with a phagosome because it is full of digestive and caustic enzymes, which break down whatever's in the phagosome, whether it's a bacterium, whether it is a bit of this membrane. So they fuse with these lysosomes to form the phagolysosome or the secondary lysosome. And the reason you can tell a secondary lysosome from a primary lysosome is it has additional structure. Primary lysosomes are usually homogeneous. Secondary lysosomes generally have various components to them. Some will be more osmophilic or blacker than others. In the case of a phagolysosome, which is combined by a primary lysosome with one of these multivesicular bodies, membrane generally can't be broken down all the way, okay? And it forms these granules here within the phagolysosome or the secondary lysosome, which we recognize as lipofuscin. If we go back to what we know about light microscopy, we know that as a cell wears out, as a cell ages, the cell generally will build up lysosomes which contain lipofuscin. We don't see the lysosome on light microscopy. We do see the lipofuscin granules. And lipofuscin granules are generally a granular brown material, and they are the final breakdown product of cell membranes. Now in this secondary lysosome, the cell has two choices. It can just hang on to it for the lifespan of the cell, and we do see that in some cells, especially neurons, or it can spit it out into the extracellular environment. But the bottom line is this secondary lysosome, full of lipofuscin, will never completely digest that. Good, so to review, when we look at this particular electron micrograph, we see primary lysosomes, membrane-bound, generally homogeneous. We see larger secondary lysosomes, which contain lipofuscin granules, which are breakdown product. Here we have one that has engulfed two microvesicular bodies. One a long time ago, and we have lipofuscin. One, it looks like it is just engulfed and is starting to break it down. It's in the very early stages. You have a little bit of imbibition of fluid by that microvesicular body, and eventually it will be digested. Here's one that's in the process of being digested into lipofuscin. Always we want to look at the remainder of the cell. We have these large mitochondria with very small cristae. We're probably in the liver. We have some rough endoplasmic reticulum and some other structures we'll talk about shortly. Non-organelle structures within the cell, which are of importance. We're still in the liver cell. Look at the mitochondria, very small cristae. We can see outlines of rough endoplasmic reticulum, some dilated, slightly dilated profiles of smooth endoplasmic reticulum. And a lot of that is obscured by this black granular material, which is glycogen. Glycogen is densely osmophilic, meaning it's going to take up our stained material. And you can see that you have either aggregated bits of glycogen or beta particles, and just rarely you can see some of the glycogen particles which are by themselves. And they're about the same size as ribosomes, so they can be confused with free ribosomes if you don't have this aggregated material. Something else I want you to notice, which we see, is this large non-membrane bound, as opposed to primary lysosome, which had a membrane, non-membrane, darkly electron dense or osmophilic material. And this is what fat looks like. Fat is extremely osmophilic, and in properly preserved sections, they are large, round, black globules. Now, as I said before, the cells have to be properly preserved, because in cases where the tissue has been processed, for example, for light microscopy, you have alcohol extraction, where the tissue is passed through a series of graded alcohols. And this leaches out most of the fat. So as opposed to the black round globule we have here, you may see round areas within the cell which are dark gray, light gray, or maybe white, because all of that lipid has been extracted from the tissue as part of the processing. Okay. Normally, if tissue is fixed, especially for electron microscopy, there is no alcohol that is involved in the processing. And we use fixatives such as glutaraldehyde or Michel's media. These are non-alcohol based fixatives, which are excellent for electron microscopy. We can and often do ultrastructure analysis of paraffin blocked material. Okay, and the loss of staining of lipid is one of the trade-offs, because most of the lipid is extracted. Notice once again there are no membranes surrounding lipid. It floats free in the cytoplasm, and what happens is it often will just push material out of the way. Here we have a bunch of glycogen that has been pushed and is just on the outside of this fat globule. I wanted to just jump back for 1 second and talk just a little bit more about glycogen and fat. Glycogen and fat normally will be interspersed among all of the organelles as we see here. But as you tend to accumulate, because it is denser than the organelles themselves, it will push them out of the way. So an animal with a tremendous amount of glycogen, as we will see later on, the organelles will be peripheralized, or fat. We know that fat globules in light microscopy, large fat cells, have a nucleus and cytoplasm which is peripheralized to the edge. So this is dense material and will displace organelles. I want to look at some of the structures that we talked about previously. This is scanning electron microscopy, a freeze fracture through the microvillar border of an epithelial cell. And you can see all these projections here. And I want you to notice all these little filaments, that go up to the top of the microvillus and project downward into this tangle of microfilaments. And these are actin filaments. Actin filaments are not contractile. They simply lend structure and fullness and fill out these microvilli. Okay, microvilli do not have the capacity to move on their own like a cilium does, because cilium are made of different material. These actin filaments they're all anchored into this meshwork where they combine with some other filaments such as ankyrin and spectrin. Okay, and this is known as the terminal web. On transmission electron microscopy, you can see the actin filaments at very high magnification. They're extremely small, maybe 25 angstroms in diameter. They're the smallest of the microfilaments. And here's the terminal web where they all project down into it, and it forms a condensation down at the base of the microvilli. Remember, the purpose of microvilli is to increase absorptive or secretory area of the epithelial cell. Just another picture, longitudinal and cross section through these microvilli. You can see that we are at the junction of two epithelial cells. So we have the tight junction right here. I want to contrast that with a structure that is recapitulated many times over in nature. And we're going to see this over and over again. As we said before, all epithelial cells have microvilli. Only a small number of epithelial cell types have cilia. Okay, here are cells that have both microvilli and cilia from the respiratory tract. Cells that have cilia are generally the respiratory tract, the reproductive tract in the fimbria and the vas deferens, the ependymal cells, and there are a few others that we will look at. Now, as opposed to the non-motile filling with actin filaments, cilia are composed of microtubules, and microtubules have the ability to polymerize and non-polymerize, and this allows them to slide against each other, generating actual motion. And when we look at cilia, they have a very characteristic center. And this is composed of 9 outer doublets, 2 inner singlets. This 9 plus 2 arrangement is present throughout the cilia. Here we are cross sections, and you can see that the structure is all the same. Depending on where you cut it, it might look slightly different. It's always 9 around the outside and 2 singlets. In transverse section, you can see some of these microtubules throughout, including the 2 singlets right here. Now, there are conditions in which cilia are improperly created. This is a case of ciliary dyskinesia from a cat that was published a number of years ago in veterinary pathology. And as I say in many of these video tutorials, birth defects happen in bunches. You rarely get one by itself. Some may be compatible with life, probably the majority are not. And so we have loss of or fetal death. okay, of the most severe, or stillborn animals, or animals that die shortly after birth. But sometimes we get some where the animal does pretty well. Well, here's a condition in which we have some normal cilia, which have 9 doublets on the outside and 2 singlets. But if you look carefully, you have some that have 9 doublets and no singlets, some that have 8 doublets here and 2 singlets. So these cilia not only are malformed, but they are incapable of movement when you get away from that 9+2 configuration. It's known as ciliary dyskinesia. These animals generally die of pneumonia because their mucociliary escalator does not work properly. The cilia do not beat to push pathogens and inhaled material back up from the lower airways. And it is also associated with what's known as situs inversus. In these particular animals, the arrangement of the organs of the body are reversed. So the spleen and the stomach, instead of being on the left side, are on the right side. So a good example of how birth defects occur in bunches. As we said before, the 9+2 arrangement of the cilium is seen over and over in nature. And when cells are in mitosis, there are structures that control the microfilaments that attach to dividing chromosomes, which are seen only during the time of mitotic division. And this is known as a centriole. And a centriole essentially looks just like the base of a cilium. You have these doublets and singlets in the center. As you cut a cilium toward the base or a centriole, they tend to take this sort of elongated appearance. But I guarantee you it's 9+2. And these centrioles are essentially hooked into all of those microfilaments. And you have them at right angles to each other, one for each dividing cell. And as the cell divides, we're looking at the centriole here, and you can see at the point of metaphase, you can see the actual microtubules which are attached to those dividing chromosomes, and they're being drawn back by the centriole. So it's controlling those microfilaments or microtubules. Okay, here we are again. And microtubules are much larger than actin filaments. And I think I told you before that the actin filament is 25 microns. That's wrong. It's actually 6 to 8. And the microtubule is 25. And they're too small to see on light microscopy. But if you cut a cell at a certain point, you may get lucky and you may be able to see these microtubules. And microtubules are extremely important in other ways other than simply dividing or controlling cilia. I liken the microtubular or microfilament skeleton of the cell as sort of a train track. And organelles are constantly in motion around the cell. Okay, they have to have a way to get from here to there. Probably the best example are lysosomes and phagolysosomes in inflammatory cells, which move to the surface of the cell and back on a regular basis. But essentially all organelles, they're not locked into one particular place. They tend to roam a little bit. And certain organelles, for example, mitochondria, will organize themselves along the area of the cell that has the highest metabolic requirement for ATP. And the way that organelles move around the cell is due to these microfilament or microtubule networks, also known as the cytocavitary network. And the organelles are attached to the microtubules by actin filaments and they move along this network of filaments to different positions in the cell. This is why when you administer colchicine to cells, which causes disassembly of microtubulin, all of a sudden the cell becomes paralyzed. This inflammatory cell cannot make pseudopods. It cannot engulf bacteria and digest them. So these are microfilaments, which are made up of microtubules and are an essential component of cilia. Well, we're almost done with the cell, the parts of the cell that you need to know. Let's go outside the cell for the remainder of this section. And we are looking here at outside the cell. We have these 64 nanometer filaments. Here they're quite densely packed. And note that they have a periodicity. They have alternating lines of light and dark. And within those, we have even smaller lines of light and dark. Okay, and these are collagen fibers. Their periodicity is 64 nanometers. So it's a very regular light, dark, light, dark, light, dark. Okay, and nothing else looks like that. There are many types of collagen. This is pretty standard garden variety collagen that you will see. But occasionally, in some parts of the body, like the cardiovascular system, you get this sort of odd-looking zebra body type collagen. Collagen is one type of extracellular filament. Second type of extracellular filament, which also has periodicity, as we can see down here, okay, is fibrin. The periodicity of fibrin, the periodicity of collagen are very different. In fibrin, the lines appear much closer, 23 angstroms on average, or nanometers. And what I want you to take a look at is, look how dense this material is packed. It's just these mats of fibrin. And if you think about it, you need something when you have rupture of a vessel to stop fluid loss and stop cell loss. And you want something that's packed very close together. So this is what fibrin looks like. These dense mats of black filamentous material, much smaller than collagen. Finally, the last filamentous material is the extracellular milieu that I want you to be able to identify is amyloid. Okay? And if we look, we have this widely spaced amyloid. That's not fibrin. That's not going to stop hemorrhage or fluid loss from a leaky vessel. Okay? It is sort of a amorphous, haystack appearance filament. If you look down here, notice amyloid has no periodicity. Here we're looking at collagen, much thicker, and it has that distinct periodicity. And this is what amyloid looks like on electron micrographs. This is one of those things that looks very different than it does on light microscopy. But for the most part, let me reassure you that things on ultrastructure are fairly easy to correlate with what we see on light microscopy. Well, we have looked at all of the organelles and intracellular and extracellular components that I think you need to write a good ultrastructural description. So think about how many of those you had never heard of before. There's a good portion of the audience out there who have heard of all these things, so I'm not teaching you that much of a new vocabulary. What I'm trying to do is build your confidence here. Well, we said the first thing in your description are going to be normal findings, normal organelles that confirm your diagnosis of a particular tissue. When we talk about liver, we talk about abundant glycogen, because glycogen is only found in a few cell types, skeletal muscle and brain being other significant glycogen accumulators. We talked about and I'm going to reinforce this, the fact that organelles tend to tell you a lot about the cell type you're looking at based on the location, based on their numbers. And I think we're going to cover that. All of this fits together, and I hope you will start as we go farther along to realize it all fits together very nicely. Everything is explainable. One of the things I tell my residents is that nothing in nature happens randomly. Okay, all of this electron microscopy makes sense. It makes good evolutionary sense. Things attain the best possible layout over millions of years. And there's a reason. So if you think about why something looks a certain way, you will probably come up with a very good reason as to why it does. Well, we talked about normal. Now, the second thing in your morphological description is going to be the presence or absence of cellular degenerative changes. As we said before, you can't read necrosis because it's just black. It's amorphous black osmophilic material, a mass of congealed proteins. And it just looks like a big black blob. So the vast majority of the EMs that you will do are based on degenerative changes. Now the good thing is that all cells, regardless of whether you're an epithelial cell, regardless of whether you are a mesenchymal cell or a round cell, essentially degenerate the same way. It's a very stereotypical process for the most part, and pretty easy to understand. So let's take a look at our prototypical epithelial cell again. Cilium and microvilli on the left, basement membrane on the right. We can see that this cell has a prominent nucleus with abundant chromatin, both euchromatin and heterochromatin. We have desmosomes, as we would expect. It sits on a basement membrane. And we have large mitochondria, which appear to be oriented toward the base of the cell. If I had to pick something, I'd probably say this might be, well, it's very difficult because it has a cilium. It's sort of a combination of proximal convoluted tubule of the kidney and maybe respiratory. And we see the other structures we talked about. We talked about the multivesicular bodies. We've talked about the Golgi apparatus and the nucleolus. So we're familiar with all the organelles. And what I want to do here is I want to shock this cell and begin the process of degeneration. And the best way to do that is to shut off energy, and the best way to prevent the cell from making ATP is to give it no oxygen. And what we're going to do here is we're going to remove an oxygen supply from this source for literally a couple of seconds, and that's all it takes to start the acute phase of degeneration. The cell has no oxygen. It will begin to degenerate immediately. It will convert the ability to make ATP from oxidative phosphorylation to anaerobic glycolysis, but degeneration starts almost immediately as well. And so we've done that. Did you notice any difference? Gone from this to this. Because everything looks a little darker. Okay, within the nucleus, the chromatin is starting to clump a little bit. Let's go back. Okay, so it's very difficult to tell the difference between euchromatin and heterochromatin. Everything's starting to clump. But the initial primary change, I want you to look at this. Nucleus is in the same spot. Cilia, microvilli all look good. Junctional complexes, all the mitochondria, everything looks just about the same. But what we see is that the endoplasmic reticulum, both rough and smooth, is dilated. Let's go back. Normal, slight anoxia. The first thing that's going to happen is that those sodium-potassium pumps, which are ATP driven and keep fluid out of the cell, essentially they keep a sodium out of the cell. They're going to break down. And sodium will begin to leak down the concentrated gradient from the extracellular fluid into the intracellular compartment. And it will also leak into the endoplasmic reticulum. And remember we said the endoplasmic reticulum it joins the nuclear membrane. It's all in big parts. So when we see if it's true dilation of the endoplasmic reticulum, because these sodium pumps on the wall, are preventing sodium, they're keeping sodium out, basically you let them in and fluid's going to follow. They're joined, so the nuclear membrane is also dilated. A lot of times you'll be looking at the electron micrograph and you'll look and you say, is that endoplasmic reticulum dilated? The best way I know, the best corroborative evidence is to look at the nuclear membrane. If it's dilated as well, that is a true finding and not something that you're overinterpreting. So within seconds of anoxia to this cell, we have changes in fluid concentrations, and the cell, initially that's manifested within the endoplasmic reticulum. Very shortly afterwards, fluid will actually come across the cell membrane into the cell. But everything else looks good, except these endoplasmic reticulum, these profiles are dilated. If we look at that on transmission electron microscopy, this is exactly what we're looking at. We see all of these dilated profiles of endoplasmic reticulum. The cytoplasm itself looks darker. Well, when your endoplasmic reticulum dilates, but the cytosol itself does not, it compresses that cytosol, and so what's normally in there is compressed together and appears and assumes a much darker appearance. For corroborative evidence, the plasma membrane is also dilated. So this is a true finding. A couple of other things that may clue you into what cell type we're in. We have numerous lipid globules, and look at all of the smooth endoplasmic reticulum, which is especially prominent right now. So yes, we are in the liver. So this is acute, reversible degeneration. If you reestablish oxygen, this cell will probably carry right on. It may, for a little while, have increased amount of breakdown. It's going to replace all the lipid that was damaged, but it will survive. Let's take that another step and look at the changes that have occurred. Starting at the top of the cell, you can see we have lost our cilia, we have lost our microvilli. I call this this is important terminology, loss of surface substructures. And that entire membrane is just sort of wavy, it's just sort of there. Note how the endoplasmic reticulum is still dilated, but the cytoplasm around it is more lucent. This means that we not only have fluid shifting into the endoplasmic reticulum, we actually have a fluid shift into the cytosol as well. And if I could redraw this picture, I would show the size of the cell starting to bulge. The area, the volume is greater than normal because of the imbibed water. And that cell will bulge and it will push on cells right next to it. Other obvious changes would be the continued dissolution of chromatin. We have a lot more, bad chromatin, devitalized chromatin that appears around the outer edge or the inner side of the nuclear membrane. Notice that our mitochondria, which normally live down here at the base of the cell, are now floating free, and some have floated all the way up to the apex of the cell. And the reason for this is the same reason that we've lost our surface substructure and this surface membrane is thrown into these large folds. And that is because another early sign of acute cellular degeneration and hypoxia is the loss of the cytoskeleton. To keep all of the actin filaments bound to the spectrin and chitin and terminal web, to keep microfilaments polymerized, you have to have oxygen. That is energy dependent. Take away the oxygen, the cytoskeleton will break down, and we will lose the shape of the cell, we will lose the cell structure, we will lose what maintains the organelles in a specific location of the cell, and they will start to to float freely within the cell. Another change that you will see, that you can see easily in this electron micrograph, is look at the appearance of the mitochondria. The cristae are wider than normal. Let's go back. Here are cristae. They are dilated because that mitochondrial membrane also keeps sodium out. So when you get sodium inside the mitochondria, those cristae are going to swell first. And because they swell, they compress the matrix becomes dark. This is a change that's known as low amplitude swelling. And it's one of the last signs of reversible change. And this is what it looks like. You can see that the cristae are markedly swollen. They have pushed the matrix, they've compressed the matrix, which has become dark. Other things that you will see here, if you look, the endoplasmic reticulum is markedly dilated and irregular here, and even the cytosol. The cytoplasm looks lucent, especially up in here. It looks very lucent. It is a very fine gray color, and this is cytosolic swelling, cytoplasmic lucency, whatever you like to call it, but it is the next stage beyond simple dilatation of the endoplasmic reticulum. You have fluid, which has been taken up by the cell due to the failure of the sodium-potassium pumps. We're going to take it a step further. We see the changes we saw before, loss of surface substructure. We see cytosolic lucency. We have our low amplitude swelling mitochondria, and then we have this change. Okay? What's going on here is that mitochondrial membrane has become so devitalized, it is allowing large amounts of sodium, and this mitochondria is just totally swollen. The matrix is swollen. This is high amplitude swelling. This is the first irreversible change that we have seen. And when you get high amplitude swelling, you get membrane breakdown. You get splits in the membrane. And normally the membrane around the mitochondria, the membrane around the nucleus, membrane around the cell are lipid biolators. They have a lipophobic and a lipophilic side. And normally that's kept very regulated. Okay? The lipophobic sides oppose each other. They don't stick. But if you take the lipophilic and the lipophobic side, and the hydrophilic and the hydrophobic side, not lipo, but hydrophilic and hydrophobic, and you expose them to each other, they stick. And when you get devitalized membranes around an organelle around the cell body, you will get these lamellations of membrane, sticky membrane, which wind around something and form these sort of lamellar stacks. And that is another feature of high amplitude swelling in the mitochondria. We can see that our chromatin is pretty much gone. It is dissolved. It's dissolute. Okay? We don't have any heterochromatin, and the nucleus itself is starting to swell. About the only thing that's still holding on are these desmosomes, because they're tough, sturdy things that are composed primarily of keratin microfilaments. And keratin is fairly hydrophobic itself. This is a nice picture of the high amplitude swelling. These mitochondria are nestled between dilated smooth endoplasmic reticular profiles. And you can see that the mitochondria are swollen, and they have large numbers of these black calcium granules. On the cartoon, the end stage, all of our mitochondria are in high amplitude swelling. We have these massive granules of calcium known as woolly bodies that tell you that mitochondria is pretty well dead. The influx of sodium has become so great that you are starting to have ruptures in your cell membranes, ruptures in your nuclear membranes, as well. One of these desmosomes is still holding on, one is broken down, and it even looks like the cell is lifting off the basement membrane as a result of loss of the hemidesmosomes. But once you start to see breaks in the cell membrane, as we see all around this cell is dead. It is degenerate. This is non-reversible degeneration, and all that's left is it to shrink, to become a congealed mass of protein, and to imbibe osmium, and we won't be able to see anything at all. That is the very specific process of cellular degeneration which leads to necrosis. And for many years, that is all we considered. Within the last 20 years, a concept which was first identified back in the late 1800s, believe it or not, has come forth, known as apoptosis. Apoptosis is a form of necrosis, and people will debate that. Some people call both processes oncosis, or oncosis versus apoptosis. But to me, it's necrosis and apoptosis. And apoptosis is different in terms of a mechanism, and it's different in terms of its appearance, especially on ultrastructure. Apoptosis is what happens in a number of cases to cells that are normal for the body which do not need to be maintained, such as the endometrial lining in females, if gestation is not going to occur, or even after gestation. These cells that are there for a little while and gone, such as cells of the corpus luteum. Okay, cells of the lymph nodes. Quite a few are undergoing apoptosis at any time because they've been sensitized to a certain antigen. The antigen has not been there and they disappear. There are also a number of processes within the body, especially those which are used by T cells or cytotoxic T cells, which induce apoptosis in susceptible cells, especially neoplastic cells. Apoptosis, and this is not going to be a Gen Path lecture, is generally run by the caspase cascade. And caspases are enzymes that have very specific functions in the cell. One of the things that they do is they cleave DNA at certain points. They cleave membranes. It's a 9-compound cascade of when the liberation of caspase 9 results in necrosis of a cell. You can also see the initiation of apoptosis when you have calcium within mitochondria, or you have excessive calcium or other compounds such as copper within the nucleus. A number of things will cause this cascade, including the binding of the Fas ligand with a Fas receptor on a target neoplastic cell. And you can look, there's tremendous amounts of information available, this even in basic textbooks, and I will leave you to your own devices to learn that material. What I want to do is I want to compare and contrast what necrosis looks like and what apoptosis looks like ultrastructurally. And the basic concept of apoptosis is the cell simply breaks up. During apoptosis, DNA will be cleaved into 180 to 220 base pair segments. And you'll get this characteristic clumping of DNA in crescent forms around the nuclear membrane. Now the next stage is that the cell begins to break up. Phospholipases within the cell membrane are exposed to the extracellular milieu. The cell will begin to form little blobs, and the nucleus itself will break up. Notice that the nucleus still has these characteristic crescents within it, But the organelles themselves, the process happens fairly quickly. We do not have the protein congealing that we do in necrosis. So the organelles generally look very good, but the cell breaks up into little blebs or apoptotic bodies. Okay, some may have nuclear fragments, some may not. Some may have lots of ribosomes and nothing else. But the cell is breaking up into these small fragments, which are generally, in the end stage, eaten up by macrophages or other phagocytic cells. And this is the process of apoptosis. We're looking at a number of cells here, and this cell looks very much like the cartoon we looked at. Okay, this is apoptotic cell in a lymph node, and you can see that the nucleus has broken up into portions. Each one has this crescent here and here, and the cell is breaking up into apoptotic pieces. The organelles actually look pretty good. Let's look at another one. Okay, rot. This is a apoptotic cell in prostate cancer in a rat. And you can see that the cell actually looks good. You can see the rough endoplasmic reticulum. It looks great. Maybe here's the mitochondria. Here's some lysosomes, secondary lysosomes. That nucleus is fragmented into several pieces, and you have crescent formation of that 180 to 220 base pair fragments of the chromatin. Just one more to show you what happens, a case of human Burkitt's lymphoma, but we're looking at a macrophage. The macrophage has ingested several apoptotic bodies. This one a little earlier, this one is a little later. They are present within phagolysosomes. And if you look, you have this two pieces of nucleus here and here with a nice crescent. You have large numbers of ribosomes and rough endoplasmic reticulum. Here, everything is starting to become darker, become digested, broken down, etc. It's very difficult to make out what actually is going on. This is what happens in apoptotic lymph nodes, apoptotic anything, is the cell breaks into pieces, and usually these are phagocytized by macrophages or other resident inflammatory cells and digested. As I said before, the characteristics of cellular degeneration do not vary much between cell to cell. And if you've seen a liver cell degenerate, you've seen a neuron degenerate, and you've seen a retinal rod or cone degenerate. They all essentially do it the same way. They may do it at different rates, but they're all done approximately the same way. Same thing with apoptosis. Well, let's continue to combine some of these concepts. And I've told you, given you the ways to identify particular organelles in a cell. We need to take the next step of being able to look at and analyze the particular organelles in a cell and to decide which tissue we are actually in. Now, over the years, I've decreased this section a bit because it is apparent that a lot of people do not use EM for everything. That's an understatement. And the exam committees of the ACVP and ECVP, I believe, are a very practical and pragmatic bunch. And conventional wisdom is that if you're going to see electron microscopy on the board certification examination, it won't be something that's totally obscure like a corpus luteum, or it will be probably gleaned from the areas that we still use electron microscopy today. Okay, so I will concentrate on certain organ systems. I will probably skip the reproductive tract almost in toto. Some of the other things that we don't, the eye, so we don't use much EM there. And try and focus on systems where it's still being used, like the kidney, endocrine system, GI system, respiratory system, and very specific, commonly imaged tissues. Now, one of the things I try to teach my residents, and I will try to impart to you over the course of this lecture, is that form follows function. As we said before, nothing happens overnight. Everything takes millions of years. But if you know the distribution of organelles and you know the location of organelles, even if you're not sure what cell type you're looking at, it will help you come to an opinion about the function of that cell and perhaps what organ system you may be looking at. Now, we're looking at a cartoon representation of a parietal cell from the stomach lining. And parietal cells are cells which make hydrochloric acid. And one characteristic ultrastructurally about parietal cells is the fact that you have this network of caveolae, they're known. But essentially what it is this is the extracellular milieu. Okay, this is outside the cell. It looks like it's inside the cell. It's outside the cell. We have these microvilli which are projecting into that. And when you measure the hydrogen concentration in this hydrochloric acid producing cell, and you measure the concentration of hydrogen in the cytoplasm, and hydrogen that is concentrated here to be liberated when the body needs hydrochloric acid. The concentration of hydrogen atoms here in these caveolae is approximately 1 million times hydrogen concentration within the cytoplasm. Well, how does it get there? How does it get from here to there? Well, this is an energy-dependent process, and there are pumps which actively pump the hydrogen ion into this extracellular space, which is inside the cell, okay, and prevent it from diffusing back. And those pumps take energy. They have incredible requirements for energy to maintain a 1,000,000-to-1 concentration gradient. And so this is where you will find the majority of the mitochondria. The mitochondria live in the parts of the cell that have the highest energy requirements. So, form follows function. Where you require energy is where you will find mitochondria. Cells with a high energy requirement will have mitochondria with abundant, robust cristae. Here is a transmission electron microscopic presentation of that cell. And you can see here, I want you to ignore this helicobacterium, but you can see here your alveoli, here they are here. We only see a little bit, but around this area, you see all the mitochondria. They're also here as well. And all the mitochondria are congregated around this area to provide the energy needed to maintain that steep hydrochloric acid gradient. This is the other component of the stomach lining. We looked at parietal cells. These are chief cells, and chief cells are digestive cells. They make and package digestive enzymes. They spew those into the stomach lumen to help digest protein. This chief cell, and as we said before, form follows function. So this cell is replete with these large membrane-bound vacuoles, which are filled with digestive enzymes. Look at the base. These are displaced in the nucleus to the base, but what I want you to see is at the bottom of this cell, you have large amounts of rough endoplasmic reticulum, just stacks and stacks of rough endoplasmic reticulum. Let's put this thing all together now, okay? Granules at the top, rough endoplasmic reticulum at the bottom. Why we have rough endoplasmic reticulum? Because we are producing protein to be secreted from the cell. What color is that rough endoplasmic reticulum? Well, that part of the cell is going to be dark blue or purple. So when you look at these chief cells, the apex of the cell will be pink because the protein and the digestive enzymes, and the basement or the basal part of the cell will be very bluish. Just for completeness' sake, this is what the third cell type looks like. It's mucus secreting cell, and we have large clear vacuoles. Mucus is generally also leached out during processing. So, and those are three cells. Let's move a little farther down into the GI tract, and we'll take a look and see if we can figure out what's going on here. What cell type? Well, obviously, an epithelial cell. Look at the microvilli. No cilia, but they have a prominent microvilli. Now, here's something that's very important. Microvilli can be all the same size, as we see here, or they can be variably sizes. Now, when the microvilli are all the same size, it almost looks like the top of the cell was mowed with a lawnmower. When they're all the same size, you are located somewhere in the GI tract. And villi tend to be longest at the apex, microvilli tend to be longest farther up in the tract, in the duodenum and the proximal jejunum. And the farther you get down the GI tract, the shorter the microvilli will be. We are probably looking at multiple cells here, although the tight junctions are not really apparent. And we may be looking at beginning of one right here. Remember that when the sectioning in electron microscopy is incredibly thin, so you don't always get everything you want to see. So that's why it takes so long to take good electron micrographs, because very rarely do you have all the components that you want to see in the appropriate photomicrograph. What else do we see in here? Whoops. Well, I see large amounts of these round, non-membrane bound black globules. And we talked about this before. We see something that's round and it's dark black and it's in the cytoplasm. We want to think of fat. And this is an EM from a rat that just had a high-fat meal. And I want you to take a look right here. And probably when I said they were non-membrane bound, you said, oh, that's crazy. I can see a membrane right there, and I can see a membrane here. Well, actually what you're looking at is you are looking at the fat, which is in smooth endoplasmic reticulum, which is where it will be partially broken down. Okay, you can see that very nicely right in the center of the picture where we have a number of fat globules within the endoplasmic reticulum. So it is membrane bound, but the fat itself is not. The membrane that we see has a lot of space, and that's the smooth endoplasmic reticulum. It will be absorbed, it will be broken down. And if you look at the mitochondria, you can see that this process takes a fair amount of energy because those cristae are pretty robust. You'll also see a lot of mitochondria at the bottom where it will be excreted from the cell. That's also energy dependent. Notice how there's almost no mitochondria at the top because the absorption does not take much energy at all. Just a couple of other things that you might see. This is a picture that was published a number of years ago in VetPath about M cells of the rabbit. These are squamous epithelial cells which have a antigen presenting capability. Underneath that, you will see macrophages and lymphocytes. Things to take a look at. This would be, because the microvilli are all the same size, we would be in the GI tract. So those are M cells. Whenever I look at EM. And I've alluded to this a little bit. I try to think about what stains blue or purple on H&E and what stains pink. And as we said, the vast majority of protein stains pink. Crystalline protein, stains pink. Ribosomal protein stains blue. So many of the granules that we see will be, especially if they are dark, very dark gray or black, will be very bright pink. And areas of rough endoplasmic reticulum will be purple. Notice that these are membrane bound as opposed to fat. Okay? And the intestinal epithelium is not a cell type you would expect to have tremendous amounts of large fat globules. So this is a panicell, and not that I think you especially need to know what panicells look like. These are cells that are part of the nonspecific bacterial defense mechanism of the intestine. They store defensins, which attack cell membranes of various bacteria. But this is what it looks like on EM, and this is what it looks like in real life. We are in the colon of a chicken. And I don't know why you would want to really be in the colon of a chicken, but here we are. One thing about avians is they often have a very prominent terminal web. This is the terminal web here with the big T on it. In real life, they generally don't have a letter superimposed. But here's your terminal web. It's very prominent. This is where all the actin filaments project into. You have a couple of other intermediate filaments here. The microvilli are flush, which means we're in the GI tract, and this is another animal which has eaten a high-fat meal. I'll give you a hint. We are still in the gastrointestinal tract, which includes accessory glands and the liver. I want you to think about this. Okay, we have those round membrane-bound granules at the apex. The base of the cell has abundant rough endoplasmic reticulum, so that would probably be purple. We have a basally located nucleus, and these cells are oriented around a lumen with some very small microvilli. Have you figured out what this cell type is? Sure, we're in the exocrine pancreas. Look, the apex of a cell is full of pink zymogen granules, and the base is full of rough endoplasmic reticulum, which makes it purple. Good job. Well, let's superimpose something else. We just looked at the normal exocrine pancreas. Now what I'd like to do is shock that a little bit. Okay, and this animal was administered a little bit of purimycin, which causes degeneration of the pancreas. And what can we see that looks like degeneration? Well, first thing that I notice is there is marked dilation of smooth endoplasmic reticulum, and to make sure that this is not artifactual, this is real, I can also see dilation of the nuclear membrane. And that dilation has caused condensation of the cytoplasm, which makes it a little bit dark. The mitochondria look pretty okay. Look at the cell. This cell is bulging. It's bulging to one side, which tells me that it has imbibed too much fluid overall. And so we have cytosolic edema. Now, I also want you to take a close look at the granules. If you look at the granules, you'll see that they're not all the same size and shape. You have some that are abnormal, some that are very large and have very little of the protein in it. So this is acute degeneration of the exocrine pancreas in an animal that has been administered a pancreatic toxin. Puromycin would be good. Zinc would be another that would cause. Even if you look at the nucleus, instead of being round, it is indented, and you can see that there is marked clumping of chromatin around the edge. That is somewhat difficult interpretation that you need to have a good grounding in EM to make. It would be nice if we had additional nuclei in the field that we could see. But most nuclei, the vast majority of nuclei in cells are round, and this is not. Let's take that a step farther. Okay? This is just more long-lived intoxication. Okay? What are the changes? Well, we have the bulging of the cell, which is compressing cells around it. We have dilated, smooth endoplasmic reticulum right in here. Nucleus, and this is a tricky one. Okay, you want to have something to compare to. Looks enlarged, it's still round. We still have nucleoli. So I don't know if there's anything really going on with this nucleus. But then we have these structures here. These are large autophagosomes. Remember, a dying cell will try to conserve bits and pieces, bits of membrane, things that it can conserve to reuse again. And this is autophagosome. And this is what we see in reversible injury. It's not non-reversible. I do not see changes in the mitochondria. But this is reversible injury, and this is the cell trying to reuse effete or obsolescent parts of its membrane, obsolescent organelles or whatever, and reuse some of those raw materials because it knows that it's going to have to rebuild. Hepatic and biliary. I think that a lot of people like to do EM of the liver because you can sit down just about anywhere. Most hepatocytes look like the one next to them. You don't have to travel around a lot. So there are a lot of pictures out there, and they have really good organelles. They are easy to identify and great cells to look at. Now I'm gonna put the bottom line up front here and give you the be-all and end-all of knowing you're in the liver. And that is this structure right here with the big C in the middle. Okay, and this is a biocanalicculus. All hepatocytes are surround biocanaliculi at one end. And biocanaliculi has stubby projections or microvilli. These are the liver projecting into the biocanalicculus where it will secrete bile. The bile will be concentrated in the canaliculus and then will eventually go into the larger bile ductules, which are lined by epithelium, and eventually into the gallbladder. Hepatocytes are epithelial in nature. They have all the structures of epithelium, just not perfect like we saw with that little perfect cartoon epithelium. But we do have those microvilli. And right at the edge, you have these structures abutting the canaliculus, and these are desmosomes. And the last thing you really want if you're a liver is to have bile flowing backwards out of the canaliculus between cells, because bile is very caustic. So you don't want it to come out there. So you have those very prominent desmosomes. If we look at our mitochondria, they're fairly large, but they don't have a lot of cristae. They're sort of randomly oriented. So this is a cell which doesn't have a high metabolic activity. As we said before, liver cells generally live on the razor's edge of hypoxia, especially if they're in the centrilobular zone. There are profiles of smooth endoplasmic reticulum and rough endoplasmic reticulum and abundant glycogen. When you see glycogen, you basically come down to 3 cells anyhow. So, we were looking at liver, and just to reiterate, if you can see a biocanalicculus, and they're not always as pretty as this, because they're not always cut in cross-section, maybe cut tangentially, but if you can see that, you can be very confident that you are in the liver. I have backed off several magnifications. We're not focused just on the hepatocyte. Okay? A couple of things I want to mention. We're looking at the hepatocytic architecture. And you can, from what you know about looking at cells in the liver from 400x or high dry magnification, this looks very much like what you would expect. We have hepatocyte plates, which are separated by sinusoids. These very dark osmophilic, somewhat biconcave cells are erythrocytes. I love erythrocytes when I am looking at ultrastructure. They are very characteristic. Nothing else looks like erythrocytes. They're deeply osmophilic. They don't have a lot of lipids, so they always stain very darkly. And they have these odd shapes. And it's great. Look at this biconcave disc from this dog. Okay, so whenever I'm looking at an EM, first thing I look for is erythrocytes. Then I know I'm in a capillary. Probably not a vein, probably not an arterial because we're looking at EM. But I'm looking at, blood-filled space. In the liver, that will be the hepatic sinusoid. So we have the hepatocytic plates, we have the erythrocytes, and then in between, we have this potential space here. And this is the space of Desay. Within the space of Desay, those hepatocytes have numerous small microvilli which project into there because not only are they continually sampling, every cell in the body continually samples the microenvironment, but also they do excrete some protein, into the extracellular space. So that's the space of Desay, and the space of Desay is lined by these flattened sinusoidal lining cells. And that's what this looks like. One other thing I want to mention is I want you to look at the difference in size between this nucleus and this nucleus. They're both hepatocytes, both in the sinusoids. And if you're going to tell me that this cell is sick because the nucleus is so much more swollen, I want to remind you that function of cut is extremely important in EM. And if you section right by the edge, you're going to get what appears to be a very small nucleus. If you center down the middle, you'll get a much better approximation of nuclear size. In the liver, the nucleus is generally centrally located. It's not basally located like you would expect in many epithelial cells. But in liver cells, and in most of your round cells, they tend to be centrally located. Let's look just a little closer. The sinusoidal lining, you can see the number of microvilli projecting into that space of Desay. Then we have our sinusoidal lining cell here, and we have the erythrocytes. Notice I said sinusoidal lining cells. There are some cells, once again, we're in the space of Desay. There are some cells which are large. They are triangular shaped. They have very few organelles. And these are Kupffer cells. And the characteristic feature, I'll say over and over, of Kupffer cells or macrophages of any type, or histiocytic, is the ability to form pseudopods. Most other cells, other than inflammatory cells, do not form pseudopodia. As a matter of fact, I can't think of any. But there is abundant actin filaments within the cytoplasm of these cells, which can change from the gel form to the filamentous form and actually result in the formation of these pseudopodia. Notice there are very few organelles in the cell. Once again, form follows function. Inflammatory cells are not meant to be long-lived. And a cell will carry just as much as machinery as it needs. So you don't need Golgi and you don't need a lot of other things if you're only going to be around for a couple of hours. When we look at neutrophils, you will see that although they do have a nucleus, They have almost no other cytoplasmic organelles except for lysosomes and granules. We are in the liver. We can see some bile canaliculi, which are not in really good shape, but now I want you to think this is a dog's liver, and we can see that the nuclei are generally centrally located, but a lot of the cytoplasm is being compressed. Organelles are being pushed to the periphery. There are large areas of the cellular cytoplasm which are filled with this black granular material. And we talked about this earlier. I want you to first think about what those cells might look like. As you push cytoplasm together, it tends to get darker on EM and more brightly eosinophilic. Those areas in which there is none might be lightish white. And this is a dog. So think about what those cells might look like. Think about a possibility. If you thought that they look like this, that those areas of the black granular material would be whitish, and that you would have compression of the cytoplasm, you have a good picture. And this is a case of steroid hepatopathy in the dog, resulting from the injudicious use of steroids and tremendous glycogenosis of those cells. Those cells are known as spider cells because you have these little wisps of condensed cytoplasm, which supposedly resemble spider legs. So spider cells. And a good way to get into a good mental habit, try to see the glass slide. Take what you have, what you know, and try and make it in your mind look like what you would see on the H&E. This is from a dog. Well, I told you we were in the liver, so that's no surprise. But we can see some large fat globules here and here. Mitochondria with that couch potato appearance. Very small cristae. And all those are being compressed by these numerous dilated profiles of smooth endoplasmic reticulum. Yes, you guessed it. This is another dog with chronic anticonvulsant medication. Although you can't say that's exactly what it is. Perhaps this dog had chronic exposure to aflatoxins or was a heavy drinker. But what you can say is there's profound hyperplasia of smooth endoplasmic reticulum. Now I said that we've learned all of the organelles that we're going to, and that is it's a little white lie. We're still in the liver. Look at the couch potato mitochondria. Look at the fat droplets, non-membrane bound, sort of grayed out. And what I'm looking at are large numbers of these structures here. And they look very much like primary lysosomes, which is pretty much what they are. Okay, but this is a specific type of lysosome that is present within hepatocytes when you have a lot of fat. Whether it is, generally it's chylomicra, and the liver is being flooded with fat. And these lysosomes proliferate, or these peroxisomes will proliferate in large numbers, help to break down the excess fat. And they are called peroxisomes. And peroxisomes were all the rage back 15 years ago as we were developing the first statins to control hypercholesterolemia. And this is from a rat, and rats develop huge numbers of peroxisomes, but far more than we normally see in humans. So rats were thought to be a good animal model, then turned out they developed all these peroxisomes, and they're not such a great animal model. But you can see this, and when you see this in the liver, and generally they always have that dark nucleoid, which is probably degenerate fat or membrane in them. So they're all essentially, most part, they're secondary lysosomes. But if you see that in a rat, especially with fat in the cytoplasm. You want to consider that these animals are hypercholesterolemic. We're looking at a cat liver. We have two large cells here that I want to specifically look at. And they're separated by a potential space with microvilli in them. This is actually a greatly dilated microvillus. Biocanalicculus. Greatly dilated biocanalicculus, and the granular material within is bile. And let's look at these two hepatocytes. Centrally round nucleus. Don't make the mistake say one nucleus is enlarged or one is too small, because it's a function of cut. But the thing that really jumps out at you here is the number of fat globules. Okay? This was probably paraffin-embedded tissue, or at least was not fixed for EM, because note that those globules of fat are pretty well washed out. Here's one that doesn't have any at all. And a number of these just don't seem to have any. This is hepatocellular lipidosis in the cat. You know that hepatocellular lipidosis is a chronic disease of cats. We don't know what exactly incites it, but you usually see it in obese cats. And they often have icterus toward the end. And the fact that we have a dilated biocanalicculus suggests to me that we have hepatic icterus. The cells are so swollen they are choking off these biocanaliculi. They are distended. Within the cell we have numerous aggregates of black material, which is probably lipofuscin or ceroid, okay, due to stretching and breakdown of the membrane. So this is hepatic lipidosis in a cat. Leaving the hepatocytes, they are not the only cell which is present in the liver. Hepatocyte here, those couch potato mitochondria. And then we have these cells, which are sitting on a nice basement membrane, nice trilaminar basement membrane. They have very large tight junctions. They have small stubby microvilli, which are projecting into a lumen. Okay, and very few organelles. Their mitochondria are probably the ultimate in metabolically inactive cristae. And this is biliary epithelium. Everything that you would expect. Biliary epithelium doesn't really have to do much except to line bile ducts. So very few organelles. They have minimal metabolic requirement for oxygen, although every cell does. And they have very, very large, robust, tight junctions to make sure that that bile does not leak back between epithelial cells and digest the liver proper. They are sitting on a basement membrane right here. So this is biliary epithelium. Form follows function, few organelles, tight junctions, etc. Let's move on to the urinary system. This could be quite confusing the first time that you've looked at ultrastructure. So we're going to go back to what we know about, especially the glomerulus, from light microscopy. And we'll do that by looking at scanning electron microscopy. But we are looking at the outside of the glomerulus. We are sitting on Bowman's capsule. And we're looking into the glomerulus, and you can see the convoluted outline of the glomerular capillary. And sitting on top of that are all of these podocytes. And they form a layer over the top, which is fenestrated. Okay, it has all of these little holes in between. And podocytes have primary foot processes, and these branch off to become secondary foot processes. And they cover a lot, but not all of the glomerular surface. So there are little holes here. Okay, that's okay because basement membrane is inside there. Let's look at this on EM. And before you freak out and turn off your DVD player and use this lecture as a coaster instead of a DVD, it's not that complex. To me, this is a very characteristic picture of a capillary. There's just a lot of cells. Now, first thing when you're looking at unfamiliar EM, first thing you want to concentrate on, if it's there, is the same thing that I would, and those are these structures right here. Black. We don't have a lot, but I have a couple of possibilities. Could it be fat? Well, it doesn't look like it's sitting within a cell. So that's an erythrocyte. And if that's an erythrocyte, we're looking at a capillary. And indeed we are. We're looking at this capillary, and we have plasma within it. And then we have this cell here, which is an endothelial cell. And you can see the endothelial cell, surrounds the capillary with a small amount of cytoplasm. You often don't see the nucleus like we don't see it here. We see a small rim of cytoplasm, and occasionally we see these nice endothelial cell nuclei. So that's capillary. That is our glomerular capillary. You see a basement membrane in here. Okay, right in here. Here's your basement membrane. Okay, and then surrounding the outside of that capillary are all these nuclei. These are those podocytes. Those are the podocyte nuclei, and coming off of them are primary foot processes, or podocyte foot processes. Okay, here's your it's very difficult to tell. Maybe that's the nucleus. You have primary, and then coming off of that, and intimately associated with the glomerular capillary, these are the secondary foot processes. Okay, and you can see the space between each of those foot processes. So we can filter material between them. It's not a sheet. So all of these nuclei outside here are podocyte nuclei. There's an area right here which is enclosed. It's on the inside of the glomerulus. Okay, this is the mesangium. And we have probably a macrophage, you can call it a mesangial cell, whatever you want, that is sitting there. It's very quiescent. Around the outside, we have Bowman's space, Bowman's capsule, and here is the parietal epithelial cell. Let's look just a little closer. Okay, so we have 1, 2, 3 capillary loops. We have a very nice basement membrane. Remember I said not all of them have 3 layers, 2 lamina densa and 1 lamina lucida. Sometimes they're a little more homogeneous depending on the species. So this is the inside of the capillary right down here. Here's your endothelium. Notice how it's discontinuous as well. But that's okay, because for protein to get out of the blood, it has to pass through one of these holes here. It has to pass through the basement membrane and then out through these slits, these filtration slits. Okay, and it's very difficult for large proteins to do that. Small proteins, yes. Large proteins cannot. If we do some damage to the basement membrane, which is probably the part that stops most of it, then we can have a problem. We'll talk about that in a minute. So here's our capillary, here's the endothelium, here's the basement membrane. Here are secondary foot processes and our larger primary foot processes here, which give rise to the secondary foot processes. And then we have a large podocyte. So I hope you're getting all that. And for protein to get out through, or the filtrate, it goes between the fenestra of the endothelium, through the basement membrane, ultimately out between the filtration slits. And that's a bit of a trick, the way it's set up. Now, there are over 40 different types of glomerulonephritis in humans, including minimal change glomerulonephritis. Okay, I really like to break it down to 2 different kinds in domestic species. Now, if you want to go into an entire career looking at kidneys, well, you might want to become familiar with other different types. But for the purposes of this lecture, to give you some basic concepts in the diagnosis of glomerulonephritis and how to avoid errant diagnoses I want to break it down into type 1 glomerulonephritis and type 2 glomerulonephritis. Now, before we separate those out, there are two changes that you will always see in glomerulonephritis. If you do not see them, you probably do not have glomerulonephritis. One of those is the presence of antigen-antibody complex within the basement membrane, also known as dark deposits. Okay, so you can see that within this basement membrane, which is swollen, you have all this antigen-antibody complex. Okay, thinking a little bit about the pathogenesis of glomerulonephritis, it arises as a result of high levels of antigen-antibody complex, often the result of chronic inflammation. In a dog, it might be heartworm disease, it could be leishmaniasis, it could be anything inflammatory. But you have high levels of circulating antigen-antibody complexes. And when you reach a particular level, those antigen-antibody complexes are going to go somewhere and they will precipitate out of solution. And they got to go somewhere, and normally what happens is they get stuck in basement membranes of blood vessels. And they form these particular dense bodies or dark deposits or dense deposits within the basement membrane, which thicken the basement membrane ultrastructurally, but clinically cause a disruption in the polarity, the electrical polarity of the basement membrane. And you end up with a basement membrane that is very leaky and now permeable to a number of proteins, which in the normal state would not get through. So that's number one, is the presence of dense deposits. The second finding that you'll see in all cases of glomerulonephritis is fusion of podocyte foot processes. Without both of those findings, you are not dealing with glomerulonephritis. Maybe an oversimplification, but if you take a look at this picture from a dog, you can see that these dense deposits are thickening the basement membrane. They are irregular, but not difficult to pick up. You can see the basement membrane here to here, and these are all your dense deposits. Endothelium, right here. And we do have some subendothelial edema there, but okay, so that is your typical basement membrane dense deposits, which are associated with glomerulonephritis. Now, this is very close up, so it's difficult to get a good idea of what's going on with the foot processes. This is a case of Lyme disease in a dog. And there are a couple of things going on. So this is a little complex. I want to go over it, but we're looking at the capillary glomerulus. Now, on the inside, we can see that the endothelial cell is necrotic. It is dark. It is vacuolated. It is bulging out. So I don't know, this might not be the most illustrative image that I could find of Lyme disease, but you know, obviously they want to show some certain things. Here is your basement membrane. It's very large. It goes from here to the endothelial cell. So it's extremely thickened. And you have this sort of plexiform splitting here. Okay, it is not solid. It is not trilaminar. It is split. In some areas, it is extremely lucent from here to here. And here's our dense deposit. So look how thickened this is. Here's a dense deposit. And then you have this splitting of the basement membrane, this plexiform splitting. Necrotic endothelial cell. On the outside, which is the reason that I showed you this picture, you can look at the foot processes. And here they look very good. And you can see the filtration process, and they look very nice. As we go around to the other side, you can see that they're fused. They're starting to fuse here. And to me, this is the second required component for glomerulonephritis, both type 1 and type 2. And this is fusion of foot processes. So if you're familiar with Lyme disease, it's the presence of the spirochete Borrelia burgdorferi, a chronic inflammatory disease of a number of species, including dogs, which will develop glomerulonephritis and chronic synovitis. We are looking at a case of occlusive disease in a cheetah. Here it's very difficult. We can see two large adherent erythrocytes. Okay, the basement membrane looks pretty good. It is somewhat thickened. You can see some dense deposits in here. But look at your foot processes. Once again, they're fused. Here and here. These don't look too bad. But you have foot process fusion. And it doesn't make any difference whether you have more basement membrane dense deposits or you have more fusion foot process. If you can identify both of them, you're dealing with glomerulonephritis. Now, we talked about two types of glomerulonephritis. You have type 1 or membranous glomerulonephritis, and you have type 2 or membranoproliferative glomerulonephritis. And I know there are many more like mesangiocapillary and, minimal change and all sorts, but I want to keep it very simple today. So we're just going to talk about membranous and membranoproliferative. And a light microscopy, membranous has marked thickening of the glomerular capillary walls, okay? And what we're seeing actually at much lower magnification are the presence of those dense deposits as well as the fusion of foot processes. And those taken together at light microscopic level just look like a thickened glomerular basement membrane or a thickened glomerular wall. Okay, at electron microscopy level, we can see other changes. And the difference between type 1, which is the membranous, and type 2, which is the membranoproliferative, is the proliferation of the cells. And what actually happens is, if you look at this side, we're looking at type 1. And I don't really care whether you know whether they're intramembranous deposits, subendothelial, or subepithelial. Okay, I have trouble keeping that straight. But the key between type 1 and type 2 is if you look here, the mesangial cells that live here, okay, they are extremely happy. Okay, nobody's told them that there are deposits yet. Okay, if you tell the mesangial cell that there are dark deposits within the membrane, they're going to get excited. They're going to reach their pseudopodia in and try and scoop them out and clean them up. Okay, so you have interposition of the mesangial cells. And when that process occurs, not only do you do marked damage to the basement membrane, but those cells become activated, they release inflammatory mediators and recruit more inflammatory cells into the glomerulus. So if the mesangial cell doesn't know that the deposit is there, be perfectly happy and you'll have a nice case of membranous. But if you activate it, if you clue them in that the deposits are there, they're going to not only damage the basement membrane by trying to get them out, but they are going to recruit other cells to help them. And so we are looking at a case of membranoproliferative. And the only difference here is that you have fusion of foot process. We have dark deposits in the basement membrane. Just get through here. But in your basement membrane, you also have the interposed pseudopodia of those mesangial cells reaching in to clean up the basement membrane. They do a terrible job at it. They do a lot more damage, and you end up with a more severe case of glomerulonephritis. Well, that's the glomerulonephritis story, and we're going to leave it there. but there are excellent books out there on the many forms of glomerulonephritis. Antonovich and Mastoffi's book, written in the late 80s, examines all of these ultrastructurally if you are really interested in that. We are looking at the epithelium, which lines the proximal convoluted tubules. It is classic epithelium. I want you to look at a couple of things. First, let's look at the microvillar brush border. And you can see that it is not flat. It is somewhat irregular and undulating. Microvilli vary markedly in size. And this is what you see in the kidney as opposed to what you see in the intestine, where there's variation in size. There's also very prominent mitochondria which are aligned along the deep margin because there is abundant absorption and excretion of a number of compounds, and that is all energy dependent. We push them against the gradient, and without the ATP that is manufactured by the mitochondria, we would not be able to create that concentration gradient. Okay, just looking at it sideways, we have that irregular brush border. And if you take a look at these vesicles underneath the brush border, I do not want you to confuse these with endoplasmic reticulum. These are known as pinocytotic or pinocytotic vesicles. Remember I said the cells are always sampling their environment. They need to know what's going on. They need to know in the kidney tubules, they need to know the concentration of minerals and other compounds in the glomerular filtrate. They're always sampling that. So you're going to see that, and don't confuse that with acute degenerative processes, which result in swelling. Okay, we have our large, robust mitochondria. We have our basement membrane at the bottom. And even higher magnification, look at the cristae on those mitochondria. And one way that the cell increases secretion is to provide more surface area. So we have these infoldings of a cell membrane primarily at the base of the cell. And the cell will use this ATP to push compounds across the concentrating gradient, across that membrane, and into the vessels below. So form follows function, metabolically active proximal convoluted tubules because of the large cristae, and they're primarily concentrated at the part of the cell that needs the most ATP, and that is where ion secretion occurs. Moving on, another reminder of form follows function. Moving down the nephron, we're going to skip the distal convoluted tubule and go straight to the loop of Henle. I want you to take a look at the very attenuated nature of this epithelium. Not much goes on in the loop of Henle, so there is minimal need for a lot of complicated machinery in these cells. There's almost nothing here. Basically, the loop of Henle only is there to conduct urine and to participate in the construction of a osmotic gradient. So it just doesn't need all of the cellular machinery that other parts of the nephron do. We can also see several capillaries around the outside of this image and a couple of inflammatory cells. Well, I did mention lead earlier, and these are the very characteristic fuzzy dark black internuclear inclusion that we see in animals with lead toxicosis. I believe this is a rat. We are in the proximal convoluted tubule. Look at the presence of the robust bacilli. And there's one feature here that I really want you to pay close attention to. And this is the irregular brush border that we looked at before. Okay, notice how it's not pretty like the previous picture where it just went across the top and you could follow it. This is a tangential section. And whenever I see something like this with a long microvilli. Okay, I have to ask myself, what type of cut are we looking at? And am I in the GI tract or am I in the intestine? And because, GI tract to the kidney. And because they are irregular, that is one feature that I associate with being in the kidney. The other one will be those robust mitochondria. They're generally parallel to the long axis of the cell. This is a very complex image to start from scratch, but let's see how much we've learned so far. Okay, first thing I want you to look at is this right here. We're looking at long microvilli. They are not, if we look over here, they're not all the same height, so we're in the proximal convoluted tubule. Okay, and you can see individual cells here and here. We have one here in which the nucleus is sort of shrunken and dark, and that's a cell which is probably necrotic. Okay, we see some down on this side. And here's one that's bulging into the lumen, okay, and that also may be a necrotic cell. Okay, within most of these cells, we see large, round, non-membrane bound globules. And of course, we're going to think of fat. But when I start to look at some of these, I start to see some straight lines. And fat is always round. There's very few materials in nature that actually form straight lines. Here's some over here within the cell. Here's some within the cell. And when I see straight lines, I think of crystalline protein. Crystalline protein is often very osmophilic. It would be bright pink on our H&E. And when I think about a disease of a rat, in the proximal convoluted tubule, we have tremendous amounts of crystalline protein, both free in the lumen and within the cytoplasm of cells. It brings me to a diagnosis here, which would be alpha-2 macroglobulinemia of rats. This is a problem that we see predominantly in male rats and is characterized by the presence of bright eosinophilic droplets of crystalline protein within the, primarily in early cases, within the proximal convoluted tubule. And then in later cases, or in severe cases, you can have necrosis, release of the protein into the tubular lumen, which is happening here. This is something that I just find interesting, really not of a lot of diagnostic significance, but we're looking at transitional epithelium in the bladder. And in some species, you see these small refractile, they look like tiles on the surface of the transitional epithelium, and that's exactly what they are. On scanning electron microscopy, these tiles come together to help give additional stability to the distended bladder. It's sort of a cool thing that I thought you might like to see. Let's leave the urinary system and go to another system in which you will occasionally see electron micrographs, and they can be a little bit confusing. Well, we're up in the upper respiratory system. This could be the sinuses, this could be the trachea, this could be the middle ear. And the reason we know we're in one of these locations is that these epithelial cells, along with the very prominent tight junctions, have cilia. Here are your microvilli. Remember, you always have microvilli. You only sometimes have cilia. And at the base of each cilium, you will see basal bodies. And basal bodies look very much like centrioles. They sort of anchor and control the cilia, and you do have the characteristic 9+2 pattern of microtubules that go down into the bottom of the basal bodies. Not all of these cells that line the upper respiratory tract are ciliated, and there is one notable exception here. Here we have a ciliated epithelial cell, Here are the cilia, here are the microvilli. And then we have these two cells on either side. And the cells bulge into the lumen, and they have large mitochondria, and they are replete with smooth endoplasmic reticulum. And these are Clara cells. And Clara cells are similar to hepatocytes in that they are a primary source of detoxification of inhaled toxins. Very interesting cells, have the ability to actually turn into ciliated respiratory epithelial cells from time to time, but they primarily detoxify inhaled substances. And this is a calf. This is the Clara cell from a calf who was intoxicated with 4-epimenol. And what I want you to take a moment to look for are all of the signs of acute degeneration that you can see. I can see a convoluted nucleus with nuclear chromatin dissolution and clumping around the outer edge. I can see multiple dilated profiles of smooth endoplasmic reticulum, here marked with an E. I see areas where I believe there are areas of cytosolic lucency here. Okay, here and here. The cell itself is bulging, which tells me that it has imbibed a lot of fluid. We also have loss of the surface substructure. Okay, many of those microvilli are missing. Here's an area where they are entirely missing. So these are all evidence of acute cellular degeneration, and this animal is intoxicated. Let's move into the lung, and several things I want to say here. Number one, this can be confusing because to me it brings up the possibility of two things. We have a lot of white space here, and we have cross-section of several capillaries, and we can see those biconcave erythrocytes. They're not always biconcave, it depends on how they're cut. But if they're cut properly, they will be biconcave discs, and they're always darkly eosinophilic. So we have 1 capillary, 2 capillaries, 3, 4, 5 capillaries. Okay, within this capillary we have several erythrocytes, and we have these flattened structures here, which are platelets, and then you can see a thin rim of endothelial tissue. And I believe we're seeing a cross-section of the endothelial cell. And I'm sorry, this is just jumping around on me a little bit today. Okay, here's the second capillary, and there's a large endothelial cell nucleus which appears to be bridging the capillary. I don't think it's blocking the capillary. I think that's a function of cut, and we have cut through, so it just appears that it is bridging that capillary. But we can see that thin rim of endothelium around that. There's our third capillary. There are a couple of other cells. This is our alveolus here. A couple of other cells outside of the capillaries projecting into the alveolar space that we need to take a look at. This nucleus belongs to a cell that is wrapping around the capillaries, another very thin rim of cytoplasm, and that is the type 1 pneumocyte. Okay, remember that when blood diffuses from the alveolar space, it diffuses through the endothelial cells, the pneumocyte capillary through the basement membrane, through the endothelial cytoplasm, where the oxygen can be picked up by the erythrocytes. Okay, obviously this has to be incredibly thin, as we see here. If you have anything that's thicker than this, oxygen will not be able to diffuse into the capillary and be picked up by the passing erythrocytes. The other cell that we have here is this type 1 pneumocyte. Type 2 pneumocyte. Here's type 1, very thin. Type 2s are very blocky, and they often have these lysosomes full of these lamellar zebra bodies, and that's surfactant. But look how thick this type 2 pneumocyte is. Obviously, no oxygen is going to pass through there. So this is why type 2 pneumocyte hyperplasia is such a devastating problem after type 1 pneumocyte necrosis. Because once an alveolus is lined by these regenerating type 2 epithelial cells, you don't have any air passing through it or any oxygen, and that's, alveolus is obviously non-functional at that point. Here's our type 1 epithelial cell again, erythrocytes, a platelet, and you can see all the components of the alveolar wall right here. We have pneumocyte cytoplasm, basement membrane, endothelial cytoplasm. And that's what a functioning alveolus looks like. Well, here's our friend, the type 2 pneumocyte, just a little higher magnification so you can get a good look at some of those surfactant-laden lysosomes. Well, they're not lysosomes, they would be phagosomes. And this is what a macrophage that is releasing surfactant looks like. Okay, here are your zebra bodies or lamellar bodies, and they reach the outside surface of the cell right here. Here's the cell membrane, and they are liberating their surfactant. As I said before, type 2 pneumocyte hyperplasia is devastating. Okay, this is an ox with lung damage, and even though we don't have an erythrocyte to look at, we have a nice alveolus here. This is a neutrophil, so this is where the capillary is. Okay, right here, it's very thin. And then we see this proliferation of type 2 pneumocytes. You can tell because they have the surfactant here and here. Okay, how much air do you think is going to go through this wall? It's probably a fibroblast here. Okay, not quite sure. This could be another neutrophil here. But do you think we're going to have any oxygen diffuse through this wall? Of course we won't. And so this is a nice representation of alveolitis, chronic alveolitis in an ox, and why type 2 pneumocyte hyperplasia is devastating to the animal. Endocrine tissue, a lot of it looks the same. There are a couple of times where endocrine tissue can be very characteristic. We are looking at the pancreas, and this is the islet of Langerhans. And on top we have a alpha cell, and the cells in the bottom half of the picture are beta cells. And many endocrine organs that have granules have what are known as neuroendocrine granules. Okay? And they generally are dense core granules that are membrane bound. They are most often round, and they have a membrane around them. You'll see this as well in the medulla of the adrenal gland. You'll see this in neuroendocrine tissue throughout the body, whether we're looking at the baroreceptor organs of the heart or we're looking at a carcinoid arising from the GI tract. They all, even the medullary cells of the thyroid gland or the C cells, all have these characteristic dense core granules. When you get to the beta cells, the granules of the beta cells are very distinctive. They may be linear, as we see here in this picture right here, or little check marks, but they often have straight lines. They're very abnormal. Some of them will have the normal round nucleoid, and many of them will have some that are fairly linear. Those are beta cells of the pancreas. So when you see those type of granules, you know that you're in the beta cell. And here's one from a vole, and you can see all of these linear granules. Here's one that's linear, here's one that's in a check mark, but they do have straight lines. These are very characteristic. The ones with the round dense core granules may be alpha cells, they may be any other cell, but they're definitely not beta cells in the pancreas. let's superimpose a little bit, as we have done before, of acute cellular degeneration over these cells. And what I am seeing, I don't see too many. I see a little bit of dilation of the endoplasmic reticulum. Don't see too much change, okay, here. But I definitely see a marked decrease in granules. This is a vole, and this is a diabetic vole. And you can see the remaining granules that are marked by the arrows are not very, the granules are not dense. They're sort of a light gray. They are variably in size. And you can see large stacks of rough endoplasmic reticulum. This cell is trying to make insulin. It just can't. It is depleted of granules, and this allows the rough endoplasmic reticulum, probably hyperplastic rough endoplasmic reticulum, to be seen by the naked eye. Well, not the naked eye, but electron microscopy. Moving on to another organ. This is the follicular epithelium of the thyroid gland. And you can see the nucleus of the cell. You can see abundant rough endoplasmic reticulum. You can see the microvilli which project into the colloid-filled acinus. And one characteristic of thyroid epithelium is the presence of numerous secondary lysosomes. And the secondary lysosomes are extremely important for the elaboration of T3 and T4, because the T3 and the T4 have to be cleaved. Okay, there are substances which are attached to them which allow them to remain in the colloid before they can become active in the bloodstream. They must be taken up by phagosomes, which fuse with lysosomes to cleave various lipophilic moieties off them and render the active construct, which is then passed through the basement membrane into the adjacent vessel. So thyroid tissue, generally you have microvilli, colloid-filled acini, which abut that, and numerous secondary lysosomes. Well, this is the other cell which is in the thyroid. These are C cells. Okay, C cells do not abut a colloid-filled acinus. They do not have large numbers of secondary lysosomes. They have occasional granules. Usually they have a little more than this. Membrane-bound granules. And they're in small nests and packets sitting on a basement membrane. This is a C cell which will release calcitonin. There's one cell here that is quite different, and the cytoplasm is filled with numerous large mitochondria. Now, mitochondria will stain bright pink on H&E. So this cell would be seen at high magnification on light microscopy to have numerous bright pink cytoplasmic granules. And it's what we call an oxyphil cell in the thyroid. You may have heard of the term oxyphil cell adenoma or Hertel cell adenoma. And these are small tumors of these cells with large numbers of mitochondria, and the cells and the tumors stain bright pink. Take just a moment, take a good look. Can you tell me where we are? When I look at this cell, I can see that the cytoplasm contains large amounts of smooth endoplasmic reticulum with some rough endoplasmic reticulum, abundant fat, which is marked by an L. You have some secondary lysosomes, which are filled with lipofuscin, but most characteristically, those mitochondria have large cristae which have vesicular swelling, or tubulovesicular cristae. If you put all of that together, we are in one of the cells of the adrenal cortex. And it all makes sense. Okay, these cells secrete steroids, and the sterols that they make are created in the smooth endoplasmic reticulum. One of the main substrates of sterols is fat. So we have all the machinery, and we have these mitochondria with tubular vesicular cristae to give us all the energy that we need to make fat. And as we break down the lipid and make the fat, the byproducts, the lipid byproducts will be broken down in secondary lysosomes and become lipofuscin granules. Making steroids is very hard on a cell, very hard on the organelles. So you often have a lot of lipofuscin in these cells. Well, there's something seriously wrong, and we're going to see this again and again. First thing I want you to look at is the mitochondria. And these mitochondria, once again, have those tubular vesicular cristae, which are characteristic of steroid-producing tissues. In between them and the cytoplasm proper, we have numerous dilated smooth endoplasmic reticulum. And to make sure that this is not just an artifact, I'm going to have you look at the nuclear membrane, which is also dilated. It's not horrible in terms of the degeneration, but it is present and it is valid. And then we have all of these structures here, which are surrounded by lamellar bodies or pieces of degenerate membranes. It's very difficult to say exactly what those were. They were likely organelles, and this cell has been treated with some form of cationic detergent. And what detergents do is they break down those lipophilic and lipophobic membranes, and they break apart and they whirl around anything they can find, and they stick together. And here they are sticking very densely in the center, less densely around the outside where they look like onion skin. And these are probably some organelle there whose membrane was damaged by a detergent. So I can't tell you what organelles they are. I would suppose that some may be mitochondria, some may be lysosomes, and I can't tell you what type of compound it is, but it is strongly cationic because there's a very characteristic pattern of damage to these organelles. Staying in the adrenal gland, when we look at the adrenal medulla, you will see that there are very characteristic neuroendocrine granules. One of the things that most residents know, and I'm not sure why we learn it, but we always do, is that if the cell is secreting epinephrine, the granules tend to be central within the membrane. When we look at norepinephrine granules, they tend to be somewhat eccentric. So epinephrine, norepinephrine, central, and eccentric. Let's move on to the nervous system, and I want to show you the prototypical neuron. Okay, let's think a little bit about neurons. Neurons, they are long-lasting. They hopefully will last for your entire lifetime. They generally don't have lots. They require a lot of glycogen, a lot of glucose, but they don't have a tremendous amount of oxidative capacity, not like muscle, which is going to beat and require a lot amount of oxygen. So if we look at their mitochondria, we're not surprised to see that they have couch potato type mitochondria. Now, do neurons produce protein for export? Well, they do to a certain extent. They do produce some neurotransmitters, but the vast majority of the protein they produce is intended to replace the cell over time. These are long cells, they have lots and lots of membrane which wears out and needs to be replaced. Okay, all the cytoplasmic components, they need to be replaced. So when we think in terms of the ribosome array in these neurons, they have tremendous amounts of free ribosomes. And the areas in which the free ribosomes congregate is known as Nissl substance. It is densely purple or blue, as we would expect. Okay, the nucleus here, this is obviously a metabolically active cell because there's almost no heterochromatin. And there's a very nice, well-delineated nucleolus adjacent to the nuclear membrane. So this is a very nice prototypical neuron, and we have a good explanation for just about everything about the cell's appearance. Let's start in the peripheral nervous system. And we are looking at a cross-section of numerous axons. Now, you never just have an axon. You have a combination of myelinated and unmyelinated axon, and every axon along its length is surrounded or invested by a thin layer of cytoplasm from a Schwann cell. And the Schwann cells can be normal or at a distance from the axon. This one happens to be right by the axon. The nucleus is right by the axon. And you can see that two axons, maybe even a third very small one, here is invested by the Schwann cell. Schwann cells perform a nutritive and a nourishing, and essentially they are the nurses of the peripheral nervous system. We have a similar cell type in the central nervous system called the oligo. But each Schwann cell or each oligodendrocyte will invest up to hundreds of individual axons. We also have a nice fibroblast or two here. But look, every axon is surrounded by the cytoplasmic projection of a Schwann cell. Now, the vast majority of cells are unmyelinated. They are slow conduction. But especially our motor neurons have a need for rapid conduction. So what happens is the Schwann cell surrounds the axon, and it begins to twist. This one is twisting in a clockwise fashion. And as it twists, it compresses all of the cytoplasm out from between the cell membrane. And it continues to twist, and it gets tighter and tighter and tighter. And eventually what happens is you squeeze on the most innermost layers, you squeeze all of the neuron cytoplasm out of the axon, out of the Schwann cell. All the Schwann cell cytoplasm, and you just have the apposition of cell membranes. Okay, this is myelin, and we know that myelin is mostly fat, mostly lipid. That's why when it breaks down in cases of white muscle disease, it turns somewhat yellow due to liberated lipochrome pigments. But why is it all fat? Because myelin is nothing but hydrophilic, hydrophobic, cell membrane against cell membrane or against cell membrane. Okay, and the biggest content of cell membrane is lipid. So myelin is predominantly all fat. And being all fat, it is quite osmophilic. And it tends to stain very dark black. This is what makes myelinated axons such an easy thing to identify in the nervous system. It surrounds an axon, it is dark black, extremely high magnification. You can see all of these are layers of cell membrane of that Schwann cell or that oligodendrocyte, between which all of the cytoplasm has been squeezed out by the progressive wrapping of these cells. Not something you absolutely need to know, but I want to show you what a node of Ranvier is. And a node of Ranvier is what we see in a myelinated axon. And you can see that periodically along these myelinated axons, you will see these little areas where they are not myelinated. Okay? We're looking at a cross section. Here's a myelinated axon. It's very dark, and if we got close, we would see the layers of Schwann cell cytoplasm that form the myelin. And then we get to this unmyelinated area, and it starts up again. And the depolarization of myelinated axons, the nerve conduction doesn't really go down the entire axon. Okay, it jumps from this node to the next node to the next node. And this arrangement markedly enhances the conduction of depolarization in myelinated nerves. So that's what a node of RAND VA looks like, if you ever have the opportunity to see it. We are jumping from the peripheral nervous system into the central nervous system, not a big jump. And if you look at this somewhat complex slide, you will see numerous axons. And I apologize, the mouse tends to have a mind of its own. Okay, but you will see numerous unmyelinated axons, and occasionally here are a couple of myelinated axons. Okay, and just like in the peripheral nervous system, this oligo or oligodendrocyte has its tendrils around each and every one of those nerves. Most of them it wraps around once or twice, and that's enough. Okay, those are unmyelinated. But some it wraps around and squeezes everything out of its projections and forms that myelin coating. Up here in the upper right-hand corner, this huge nucleus is a neuron. Now, I don't know that much about electron microscopy when it comes to the nervous system. But I do know that I can identify a myelinated nerve, and these do not look good. As a matter of fact, the myelin is splitting, and that's what it's called. It's called myelin splitting. There is edema, which starts out very small between those layers, and then becomes very severe. So we have the axon here, and then that myelin sheath is just totally chewed up in this dysmyelogenesis of Siamese kittens. So when you see this, you know that the myelin should be very orderly, very lamellar. You shouldn't really even see the individual lamella. And here you have tremendous amounts of edema within this myelin. You can call it myelinic edema, you can call it myelin splitting, but it is a real problem. Is the axon working? Well, it probably is, but it's not working well. Just a little reminder, we are looking at ciliated epithelium in the central nervous system. You will normally see ciliated epithelium in the ependymma. It helps sort of move the cerebrospinal fluid along. Okay, so these are ependymal cells. Heart. Now it's interesting because one of the main questions I get about heart muscle is, do I need to know everything that's in the anatomy books? And there are a lot of bands, the I band and the A band and the Z band and the sarcomere. And in terms of pathology, I think if you're an anatomist, especially in these areas, it's very important to know this material. But I generally don't worry so much about A bands and A zones and all of this stuff. The two things that I do keep track of, I refer to everything from here to here as the myofilaments. Okay, they are actin and myosin filaments, which in the presence of ATP and calcium have the ability to travel along each other, allowing the muscle the ability to contract. So I call them myofilaments. Okay, on either side of the myofilaments is a very dark black band, which is the Z band or Z disc. And that's the way I remember it. Okay? When the muscle contracts, the Z bands will get closer to each other. If you look at this, the A band itself will not decrease in size. Only the I band will move. So those Z bands will come into the middle here and line up against where the greatest concentration of myofilaments are. That's how the muscle contracts. But if you can remember, myofilaments or actin and myosin filaments and Z bands, really, that is what you need to know for the purposes of description. Muscle cells have the largest and most robust mitochondria, as you can see. They're all located linearly at the edge of the muscle cell. And they also have an equivalent of the endoplasmic reticulum called the sarcoplasmic reticulum, which reaches down very close to the myofilaments. And the main purpose of the sarcoplasmic reticulum is to make calcium available for contraction. It is where calcium is stored in the muscle cell. And when it is segregated there, the muscle is relaxed. When it is liberated, the muscle will contract. So that's the sarcoplasmic reticulum, those very prominent mitochondria. Another question I get very commonly when looking at muscle is, how do I tell the difference between cardiac muscle and skeletal muscle? Well, the answer there lies in the nature of the contraction of those muscles. When you look at your skeletal muscles, like your bicep, and if you flex your arm and extend it, that contraction is the same over and over again. It's on the same plane. So skeletal muscle is generally arranged like we see in the bottom left-hand corner. Everything is in alignment to make a standard contraction. Okay, all of the myofilaments or the A band is lined up. All of these Z bands are lined up. The mitochondria themselves are lined up. Okay, everything is in register. When that muscle contracts, it's going to contract the same way time after time. Think about the contraction of the heart. There is minor changes every time your heart beats. And the contraction is based on conduction of electricity through the system. Okay? It's not the same contraction over and over. So what happens here is that these myofibers can be arranged any way they want. And so you can see that the Z band sort of meanders back and forth. It's not in line. It's not in register like it is in skeletal muscle. It just moves any way it wants like a lazy river. Okay? The A bands are not lined up. Okay? So it's a different type of contraction, and the muscle is set up a different way. Now, I'm not going to teach you all the ins and outs of myofiber degeneration. We can see this in a wide variety of tissues, including foot and mouth disease, anytime you have muscle damage, toxicoses by plants, and certain other toxins like furazolidone. In this case of white muscle disease in a calf. And muscle has basically the same type of degeneration as any other tissue with one major change. Remember we talked about degeneration, lack of oxygen, and how it is so important to keep the cytoskeleton together. Well, what is the major component of the cytoskeleton in a muscle fiber? It is those actin and myosin filaments. So when you shock this cell, when you make it unable to create energy and ATP, you will get all of the same changes you will see in other cells, including cell swelling and changes in the mitochondria. But the most important thing is the dissolution of these actin and myosin filaments, which are the cell skeleton. This is known as myofibrillolysis. And what we're seeing, this is a fairly mild case, but these muscle fibers, see how disorganized they are. These Z-bands no longer line up. This is known as Z-band streaming. And they actually go horizontally because the fibers are breaking down. We have dissolution of the actin and myosin fibers. Once again, the term is myofibrillolysis. You can see other changes. You can see abundant cytosolic edema. You can see degeneration of the mitochondria. But the key word at the ultrastructural level for the changes that we see in degenerating myocytes is myofibrillolysis. Myofibrillolysis. Let's put a couple of these together. We've seen this before. We have diffuse myofibrillolysis. Okay, you don't see that. And then you see these large organelles with lamellar bodies around them. Can't tell you what it is, what caused it, but you could say that this is a cationic detergent. So this is a cationic detergent affecting a muscle cell and causing marked myofibrillolysis. In this picture from a duckling's heart treated with furazolidone, by John Van Vliet many years ago, once again, you can see marked myofibrillolysis. There's intercellular edema, dilation of the endoplasmic reticulum, cytosolic lucency, streaming of the Z-bands, and myofibrillolysis. Sometimes the myofibrillolysis gets so bad that you simply can't see it at all. You might just see something that looks like this, but there's almost no myofilaments left. Now, as long as the sarcolemmal membrane is not breached, the animal will be able to regenerate this. It's once the sarcolemmal membrane is breached, usually by inflammatory cells who get in there and start eating everything up, it'll never regenerate. So severe myofibrillolysis. We've talked about cardiac muscle and a little bit about skeletal muscle. I want to talk a little bit about smooth muscle, because smooth muscle, once again, looks different. It looks very different. It doesn't look like skeletal muscle at all. We're looking at a smooth muscle cell, or actually sections through two smooth muscle cells. Note you do not have the large, robust mitochondria because you generally don't need it. Smooth muscle cells, we look at this cartoon here. When they contract, they don't shorten, they just round up. And that is because they don't have the typical arrangement of myofilaments that skeletal and cardiac muscle do. And their myofilaments, instead of anchoring into Z-bands, they anchor into these what are known as dense bodies or focal densities. There's a number of names for these. And this is how the actin myosin filaments run from focal density to focal density. And when the muscle needs to contract, it will pull against each other and the cell itself will round up. So you don't see the actin myosin filaments nicely like you do in cardiac or skeletal muscle. And the Z-bands are these densities. Generally, the nucleus is centrally located. It's often considered to be crimped. And then inside all of these myofilaments, you will see the cytoplasm of the cell proper with mitochondria, abundant glycogen, and here's some either smooth endoplasmic reticulum or golgi. They do have caveolae, which are the equivalent of endoplasmic reticulum in smooth muscle. Just a couple of other things in the hematopoietic system. If you look very closely at endothelial cells, you'll see some structures with this very nice lattice work. Okay, and this is where von Willebrand's factor factor 8 related antigen is found in the endothelium. But you have to be at extremely high levels. We also see a little bit of a multivesicular body in here. Looking at the wall of a blood vessel, and this is a rabbit. This is a special type of rabbit known as a Watanabe rabbit. If you're familiar with the Watanabe rabbit, you'll say, oh yes, I see what's going on. Okay, we have the wall of the vessel. On top we have the endothelial cell, right here. And then underneath that, in the tunica intima, here's our smooth muscle layers. In the tunica intima, you see this plaque-like formation with abundant, clear, non-membrane bound vacuoles, the center of which is now white, and that is lipid. Okay, between that we have concentric areas of additional lipid, and here we have some dark black mineral. And this is a case of atherosclerosis in a rabbit. So knowing you were in the arterial wall and seeing this, hopefully you would be able to pick up. There's a fair amount of mineral in this plaque as well. This is an aortic or an atherosclerotic plaque. There are very few macrophages in this particular one. It just has mineral and lipid, and more mineral in the wall of the blood vessel. Just to show you a similar picture, but this is from a primate with abundant mineral within the wall of this vessel. And this is arteriosclerosis. There are a number of types of arteriosclerosis in non-human primates and in man, and we also see it in other mammalian species. And the characteristic of arteriosclerosis is the presence of abundant mineral within the wall of the vessel. It often results from hypertension, damage to the wall. The last image for the hematopoietic section, and what I'd like you to do is to shut the machine down for a sec, take a good look at this, and see if you can figure out what's going on. It'll put together a lot of the components that we've talked about in the last several hours. Okay, well, I hope you got this one. And there are a couple of very identifiable things here. Let's start with the presence of erythrocytes. Now, I'm not sure exactly how this was stained, but for some reason, the erythrocytes did not pick up the osmium like I like them to do. Normally, they're dark black. But these sort of irregularly round shapes are very characteristic for erythrocytes, even though they are they're not the nice black ones that we're used to. So that tells us that somewhere in here is a vessel. Can we see anything else that we recognize? Well, I have this multi-nucleated cell. It's not really multinucleated, it's one nucleus, but it's multilobated with abundant granules. We're next to erythrocytes, so I think that's a neutrophil. There's actually another one up here. Okay, it's a neutrophil. And so if I have neutrophils and I have erythrocytes, I'm probably in a vessel. Okay, and so you have to look for the vessel wall. Okay, so the vessel wall comes here, and there's a break, and that's here as well. And then we have all of these here. We have one here, we have this one sort of disc-like thing, and a couple which are sitting within that break of the basement membrane. And these are platelets. Okay, and they're forming a plug. If this is our basement membrane, sitting on a basement membrane, we would expect an endothelial cell, and here's the endothelial cell nucleus. So what we're looking at here is a capillary in which there has been a break, and we have the early formation of a platelet plug. Now outside this vessel, we have this neutrophil here. Not quite sure what this is here. Maybe it's another neutrophil. Very well might be. They do tend to deform when they come out of vessels. Okay, we have some erythrocytes outside of this vessel, so we have hemorrhage. Oops. And if you've recognized this material, I showed to you earlier, it's very dense, filamentous. You can't see through it, doesn't have any periodicity, so this would be fibrin. And scattered throughout all of this, we have these round cross sections, very dark black, and they lack any real structure. And those are bacteria. And this is a case of anthrax in a macaque. And what we're looking at are the effects of the anthrax bacillus's virulence factor, both lethal factor and edema factor. We have rupture of a vessel, hemorrhage parexis with fibrin and extravasated erythrocytes, and there are probably a lot of edema fluid in here, and the presence of those bacteria. So if you identified everything that was going on in there, although you might not have identified the bacteria, but you identified hemorrhage, formation of a platelet plug, and then fibrin and hemorrhage in the surrounding tissues and mixed with bacteria, you are doing fantastic. If you didn't, you're probably still doing pretty well. But we'll continue to work and continue to put this material together. Musculoskeletal system, we have already looked at skeletal muscle, so once again, all of your myofilaments are in register, all of your Z-bands are in register, all of your mitochondria line up, so we can give that same contraction over and over and over again. Another little bit of skeletal muscle with severe damage. We have diffuse myofibrolysis, formation of abundant lamellar bodies. This is somehow some cationic agent has been injected into this muscle to result in this very particular change. Just a couple of other cell types. We are now in bone, and the dark black sort of fibrillar material around the edges is actual bone. It's mineralized matrix, and it comes out very dark black. Okay, within this mineralized matrix and separated by a clear zone is a single cell with abundant euchromatin, a small amount of heterochromatin, and numerous profiles of rough endoplasmic reticulum. This is an osteoblast, and it's sitting in its lacunae. This one's just a little different. These are osteoblasts, which have more rough endoplasmic reticulum. It's very difficult to tell the two apart unless it's actively creating osteoid, and that's probably what these osteoblasts are doing. The slightly mineralized material is likely to be osteoid, and this is bone or mineralized bone itself. But it's very difficult to tell the difference between osteocytes and osteoblasts. Okay, note that this one has a little bit more euchromatin and a larger nucleolus, or a large nucleolus, but extremely difficult. This looks very similar, except the matrix is not mineralized. This is a chondrocyte, or perhaps a chondroblast, and there's some fibroblasts at the top. So this is probably a chondroblast, which is making matrix. I'm not going to spend a lot of time on the reproductive system. It's very complex. The chance of you using electron microscopy in the reproductive system is fairly small unless you're doing tox research. But just one or two things I want to point out to reinforce what we have said before. Okay, this is pictures from electron micrograph study of sperm. Okay, and this is one sperm cell. In the top right-hand corner, top left-hand corner, we can see the head of the sperm. Basically, the acrosome, and all it contains is a little bit of hyaluronidase to help dissolve the wall of the egg when it penetrates, and the genetic material. Now what I want to do is what's more interesting to me is the tail. If we look at the bottom, you can see if form follows function, look where all the mitochondria are. Okay, they are surrounding the tail. Okay, basically that's pretty much what a sperm does, is it swims. So all the mitochondria are there, tells you that's where all the action is. And if you cut that in cross section, we get the picture in the top right hand corner, the thick dark band on the outside is that row of mitochondria. Okay, and then you have in the center, the axoneme is composed of that standard 9+2 array. It's essentially a cilium. It is the tail, and this is what sperm looks like. This very complex picture is one that hopefully you wouldn't get on your board certification exam, even in your worst nightmare. But I want you to just take a quick look at this, because this is a cross-section of a testis, and you can see structures that you can identify from light microscopy here. So even if you get something that you're not quite sure what you're looking at, don't just try and run away and hide. Try and figure it out. And what we're looking at here is a Sertoli cell, right in the center, is triangular shaped. Okay, here's more down here. This is the lining of the tubule. And then you get these spermatogonia, or primary spermatocyte. And eventually you end up with one of these spermatids. Okay, now we don't have the whole thing. If we saw up here, we'd have a lot more of those sperm like we just looked at. But this is, it looks very much like it does under your microscope. The key is you're just looking at it much more closely, but you actually know most of this stuff. These are some interstitial cells from the ovary. And the only reason that I'm showing it to you is that in humans you have these very characteristic crystals. I'm not familiar with those in animal tissue, but if you look closely, you can see those tubular-vesicular cristae, the tremendous amount of smooth endoplasmic reticulum, and those cleared lipid droplets. Okay, if I didn't tell you that this was from the ovary or testis, you could probably still say that that is a steroid-secreting cell, and hopefully give me a list of cell types which it could be. We're looking at a cross-section of the vas deferens, and you can see these cilia, much longer. These cells have both microvilli and cilia, and lying on top of them are spermatids or spermatozoa. Okay, let's look at the cells of the blood. I don't think that I need to go over what red blood cells look like, because we've seen quite a few, and as I said before, the red blood cell is your friend. Whenever you're looking and see red blood cells in electron micrograph, you're pretty sure you are in a capillary. Small cell, multi-lobated nucleus, and numerous granules. Where's the rough endoplasmic reticulum? Where's the smooth endoplasmic reticulum? Where's the Golgi body? Well, it doesn't have them. This is a neutrophil. A neutrophil's life expectancy is several hours, maybe 8 hours. Now, it does have the ability to make small pseudopods. It does have the ability to change the orientation of the cell by use of pseudopods, by the formation of pseudopods by changing gel form of actin to the filamentous form of actin. But in terms of normal organelles, they're not there. This cell is not supposed to live for a long period of time. Now, let's not get into a heavy discussion of why is it having a big nucleus if it's only going to be around for a little while. But if you look at that nucleus, look at the amount of euchromatin versus the amount of heterochromatin. It's almost all heterochromatin because it's really not going to make anything. The only thing that it makes is those primary and secondary granules, which are filled with all sorts of acid hydrolases and caustic material. These are the kamikazes of the blood system, and they're just not equipped to come back home. We can see in this early case of anthrax, you can see that the neutrophil is about to emigrate. The endothelial cells are contracting, allowing the adhered neutrophil the ability to deform and to pass through this vessel wall. This is an eosinophil. And almost never will you see an eosinophil with the bilobe nucleus so well demonstrated, because it's a function of cut. If this cell had rotated 90 degrees, you might see just 2 cuts through the nuclei. So this is a very fortuitous picture, not one that you would usually see. What I want you to look at are the dense linear nucleoids within these granules. Okay, and these are granules of the dark parts, the major basic protein. It's very characteristic for eosinophils. Note how a lot of these granules are actually discs, and they are distended by the presence of that major basic protein. So they range from a sphere to a disc. Okay, so that's major basic protein, and these are eosinophils. Here is the third type of circulating white blood cell, once again multilobulated, and we have rudimentary pseudopodia, almost no organelles. This is the basophil. Basophils are ocean-going mast cells. And one of the characteristics of mast cells is a number of the granules have lovely scrollwork. In just about any species, you're going to get this very nice scrollwork. Most are dense, but you get some of these beautiful patterns within them. And you often also have, when mast cells or basophils degranulate, they often tend to leave empty spaces where the membrane resides. What was in it is gone, and it looks like these granules just sort of pop out. Okay, that's not what happens, but it gives that appearance. So you often have some empty spaces within them. We'll see that a little more in mast cells. Here's a cell with very large pseudopods, a slightly indented nucleus, and numerous granules. And look how long these pseudopods are. The only cell that makes pseudopods like that are macrophages or histiocytes. Any related cell, whether it's a Kupffer cell or a macrophage or antigen-presenting cells like dendritic cells, something like that, okay, of this lineage, of the reticuloendothelial cell lineage or histiocytic lineage, these are the ones with the beautiful pseudopods. For the smartest cell in the body, this is the most boring cell in the body. Okay, we're looking at a lymphocyte, few rudimentary pseudopods, but look, I can see a couple of organelles, maybe 2 mitochondria at the bottom, and that is just about it. Okay, and these guys, when it comes to the immune system, they run the show. They have a very large nucleus and a very small cytoplasm, so they're like big brains. Okay, they run everything, they don't actually do much, with the exception of the NK cells, but that is what a lymphocyte looks like. This is, as you would expect, let's go through this one rationally. Okay, abundant rough endoplasmic reticulum. What does that mean to us? Well, it means that the cell is secreting protein outside of the cell. It's making a lot of extracellular protein. And all of that rough endoplasmic reticulum is going to be what color? It's going to be blue or purple. And it has a large nucleus with abundant heterochromatin. I guess it's not that complex to make antibodies, which is clumped around the outside. Okay, so we have a blue cell with a centrally located nucleus with clumped heterochromatin, sort of a clock face. This is a plasma cell. Okay, all makes sense. If we saw the Golgi here, it would push those ribosomes out of the way, it would be right next to the nucleus, and it would be that very characteristic off that we see with plasma cells. So this is what a plasma cell looks like. Nucleus looks very much like what we see with a lymphocyte, a terminally differentiated B cell, which will make antibodies. And this is a type of plasma cell. But if you take a look, you have these large dilated cisterns of dark black material with straight lines in it. This is crystalline protein, and this is a Mott cell, or a Russell body cell. A cell replete with antibody, replete with protein. For some reason, it is not releasing it. So this is the appearance of a Mott cell. If we go into the bone marrow, we will see these large cells which appear to be breaking into pieces, and that's what they are doing. The pieces don't have the nucleus is large, it is multilobated, but the pieces that break off have some cytologic organelles, some rough endoplasmic reticulum, maybe a mitochondrion or two. Then they break off in these cleavage planes. This is a megakaryocyte, and these are platelets. As we all know, platelets are anuclear, and they normally float around as small flat discs until they contact a break in the continuity of the basement membrane, and they will deform and form the platelet plug. So these are platelets, and this is another great, a lot of times when you're in a capillary, you will see these floating around. You might not see erythrocytes, but you see something like this with these prominent granules. These are probably platelets. And this is what they look like up close. A little bit of rough endoplasmic reticulum, a number of granules. Okay, and you have all of the good things you can read about in your Gen Path book, like aryl sulfatase B and SRSA and histamine and all of that. PDF, growth factor, etc. Okay, a couple of things that we'll look at really quickly we have not mentioned before. This is skin, and is a nice picture of what skin, cornified epithelium or skin looks like on EM at a low magnification. Here's a capillary down here. Here are our stratum corneum, dead epithelial cells. And you can see that here we are in the stratum spongiosum, and you can make out the outline of other cells with these prominent intercellular bridges. And they get down, and eventually as we get down to the bottom, the stratum basale, you can see these cells have a well-defined nucleus. They don't have any keratin, they haven't gone really dark on us, and it sits on a basement membrane. If we look at the stratum spongiosum much closer, the one thing I want to really point out is the presence of all of these dense keratin tonofilaments, which bridge, and here are your desmosomes. There is edema between the cells, but not enough to rupture these desmosomes. And you can see how the cytokeratin tonofilaments contribute to each of those. Within the skin, you will see cells which are filled with these little dark brown bodies. And they don't start out as dark brown. They actually start out as little things that look like American footballs. They're ellipses or ovals, and they have a nice, meshed grid work within them. And over time, they accumulate dark black pigment. And these are melanosomes, and melanocytes, as we see here, produce melanosomes, and they can actually push the melanosomes into squamous epithelial cells in some cases. But these are melanocytes. And melanosomes build up pigment over time. So they're not born dark black. Those are melanosomes. And it is something that we will occasionally use electron microscopy for in the diagnosis of melanoma. Because every once in a while you get one who doesn't read the textbook, and he does not stain, or she does not stain positive for S100 or HMB45, or maybe your stains are equivocal, and you want to find out. And occasionally we do still send some of these difficult tumors to EM to look for the presence of melanosomes. These small tennis body looking bodies are Burbeck's granules, which are seen in dendritic cells in the skin. They just look nice. They don't really mean much of anything, but that's what Burbeck granules look like. Can we put this one together? Even if it's we have something written on it? Well, let's take a look. I have a triangular cell. It has abundant rough endoplasmic reticulum, which means, right, it is excreting something from the cell. And then surrounding that, I see numerous linear fibrils with aperiodicity, which are cross sections. This is a fibroblast. It's secreting lots of collagen, which is being polymerized around it. And the presence of the rough endoplasmic reticulum makes a fibroblast. Remember I showed you some fibrocytes before, and they're very long and thin. These are a little more triangular. They've got more going on here. And they're usually surrounded by abundant collagen. Here's another fibroblast, maybe fibrocyte, but probably fibroblast, which is long and skinny and surrounded by collagen. And this one has tremendous numbers of vacuoles. And this is from a paper that was published a number of years ago on feline mucolipidosis. And it simply reminds me to tell you that EM is a terrible way to try and differentiate between all the different lysosomal storage diseases. They essentially all look like this, some lighter, some darker. Okay, anything with lipid, like mucolipidosis, you're going to wash out almost all that lipid, so the vacuoles are, in formalin processed tissue are going to be clear, but essentially it's very difficult to tell one from the other on EM. This is from a dog. And we're looking at a single cell. It has a large, clear, non-membrane bound black vacuole which peripheralizes the nucleus. And this is an adipocyte. Not an adipocyte, that's one of my pet peeves. It's an adipocyte. And it's just what you would expect. Well processed, taking up a lot of osmium. And this is what fat looks like. Now, what is your diagnosis going to be? Well, it's going to be adipocyte. Could it be a lipoma? I suppose it could. Could it be a liposarcoma? It could too. Okay. One of the things that's key about using electron microscopy is not to overdiagnose, especially when we're talking about neoplasms. It's very difficult to diagnose malignancy. It's almost impossible when you only have 3 or 4 cells to look at. And remember that turned on fibroblasts can look very much like malignant cells, both on light microscopy and ultrastructure. So make sure that you do not overstate what you see. Well, this is one of my favorite examples, maybe my favorite example of how form follows function. And we're looking at a single cell surrounded by several capillaries. And it's a single cell which is composed of a nucleus, abundant mitochondria, and some huge lipid droplets, and that's it. And this is brown fat. And brown fat is what we normally see in young animals, although there are some strains of rodents that have brown fat through their entire life. And the purpose of brown fat, which often disappears as the animal ages, is to produce heat. We see it in heaviest concentrations in the very young, newly born animal who does not have the ability to regulate its own temperature. So brown fat assists in keeping the body temperature up of a young animal. And the question is, if you're a cell, how do you produce heat? We've talked about that before. And the only way that you can produce heat is to uncouple oxidative phosphorylation in the mitochondria, switch to anaerobic glycolysis, in which one of the byproducts of the production of ATP is the liberation of heat. And so these cells do that by uncoupling the oxidative phosphorylation capability of all of these mitochondria. And the lipid is there as the substrate, which they burn during anaerobic glycolysis. So form follows function. Your cell doesn't have to be around that long. It only has to make heat. So really all it needs is mitochondria and fat and a nucleus. Okay, and that's why it looks like this. And it's how it works. Okay, everything happens for a reason. All cells are put together the way they are for a reason. You just have to figure out why. We're looking at mast cells. These are mast cells from an owl, but I just demonstrate we have a normal mast cell on the left and a partially degranulated mast cell on the right. Remember I said that degranulated mast cells, it looks like the granules just popped off. I think it's because the membrane stays, the contents are discharged, and so you still have a potential area there. You can almost even see the process occurring right here. So mast cells on electron microscopy, well, if they're degranulated, will have these large white holes. And if you look closely at high magnification of mast cells in a mast cell tumor, you'll often see a similar thing with a large number of these mast cells, especially they degranulate. If you look at a good cytology at high magnification, you will see that the simple pressure that's involved in making a smear is enough to degranulate mast cells. And many of the neoplastic mast cells you see in cytologies or aspirates will be degranulated, have these little holes in them. As we said before, the granules associated with mast cells can be absolutely beautiful. And the scroll work that you see, is fascinating. I'm not sure exactly why this happens, but it's very characteristic for mast cell granules. Spend the rest of this lecture talking about some times that you may use electron microscopy in the diagnosis of infectious agents, and we're going to end up with some neoplasms. Now, it would be fairly unlikely for you to go straight to EM without having a good idea of the agent that you're looking at. When we look at electron micrographs of infectious agents, usually they're fairly high magnifications because we have an idea of the disease we're looking at. We're looking for documentation. It would be very unusual for you to be called on to diagnose an infectious agent via a electron micrograph. However, in a testing situation, you may be given a unknown and be expected to come up with a reasonable diagnosis. Now, there are a couple things I need to talk about when you are looking at viruses. To me, and I look at a fair number of electron micrographs, a virus is a virus. Okay, they are small hexagonal structures. Usually they are electron dense. They may have a envelope, they may not, but essentially most viruses look alike. We'll look at some that are slightly different, but most of them look alike. I don't approach an electron micrograph trying to get a specific diagnosis on a virus, especially as an unknown. But there are certain features of viruses that I would ask you to pay close attention to, which will be able to narrow the scope of potential pathogens. And then based on certain things like the presence of an envelope or not, the location of the virus in the nucleus or the cytoplasm, and the size, we can come up with a fairly narrow group of viruses. Let's take a look here. And if you look at this image and you say, wow, it just looks like a lot of little black viruses. I'm with you on that. Okay, we are looking at the brain of a mouse. We're looking at a neuron, and we can see that within the cytoplasm there are a number of vesicles in which are studded by these small, about 30 micrometer viruses. Okay, there are a couple of keys here. One distinct key when trying to identify a virus in electron micrograph is the location in the cell. There are a number of viruses that replicate within the cytoplasm. Those are generally the RNA viruses. And there are a number of viruses which replicate in the nucleus, which are the DNA viruses. And if you're familiar with DNA viruses and RNA viruses as a general group, that often will help you out. We are looking at the toga virus or alpha virus, which is associated with Eastern equine encephalitis. And this is an experimental case which has been inoculated into the brain of a mouse. And the virus itself comes into the cell through the use of coated pits. Viruses are very sneaky as how they get into cells, and they usually use traditional mechanisms. They attach to normal receptors, or phagocytosed, or they come in through coated pits. This one comes in through coated pits, and it hijacks lysosomes and endoplasmic reticulum and forms these vesicles. You may hear me slip and use the term vacuoles. I don't like that very much. And vesicles is a non-specific term as well, but because it will use either, it's very difficult to say whether this was endoplasmic reticulum at one time or a lysosome. Okay, but within these, it forms viral proteins and nucleic capsids, and then the virus particles will bud off of the wall of these vesicles. Okay, so the keys here, I believe, the one key that I'm trying to get across to you is the location. We know that this is going to be one of the RNA viruses based on the fact that it is manufactured in the cytoplasm. We're looking at the endothelial cell from the heart of a baboon. Okay, you can see that these endothelial cells are resting on the basement membrane. It would be very nice if we had a erythrocyte in the lumen to give us a better clue as to where we're looking at. But I want you to see that within the cytoplasm of the cell, there are a number of very small, 20 nanometers or less, virus particles which are closely clumped together in a hexagonal pattern known as a paracrystalline array. Now we know two things about this particular virus. We know that it is an RNA virus because of its presence in the cytoplasm, and there are a number of viruses that form these paracrystalline arrays, okay, including morbilliviruses and some adenoviruses, and this particular virus, which is a picornavirus. Something else that is very characteristic about this virus is its size. Picornaviruses are some of the smallest. Until recently, they were the smallest virus particle known. And they're so small that if they didn't show up in paracrystalline array, we would likely mistake them for ribosomes in the cell cytoplasm. I believe circoviruses now are even smaller than this. But lucky for virologists who are working with this virus, they show up in this characteristic paracrystalline array. So an RNA virus in paracrystalline array affecting an appropriate species. This is encephalomyocarditis virus. This picornavirus will affect non-human primates, rodents, and elephants. Because we know it's the heart of a baboon, so it's a characteristic host in the cytoplasm of paracrystalline array, we can say with pretty good assurance that this is a picornavirus. So, location in the cytoplasm, presence or absence of paracrystalline array, and knowing the host species. That's what I call playing the vet game. If you know a viral disease in a particular host species, and you're looking at that host species, certainly you want to come up with that. We are looking at the transmission electron microscopy of feces. And this is from a pig with transmissible gastroenteritis. This is a coronavirus. Now, it's not common that you get to look at transmission electron microscopy of feces, but I wanted to put this in because it demonstrates a couple of characteristics of coronavirus particles that you can't see when they are in tissue section. One is the variation in size of these particles. Okay, unlike many other viruses, coronaviruses can vary greatly in size. They can be somewhat oddly shaped. They're not perfectly round. They are not hexagonal. Okay, and surrounding the virus particles are these small projections. Okay, and these are known as peplimers, and they're characteristic of coronavirus particles. Corona from the Latin meaning crown, and it makes sense. So in feces, coronavirus particles are variably sized, and they can be very, very oddly shaped, oval, egg-shaped, sort of squashed on one side. So they're not typical round or hexagonal viral particles. When we take a look at them in tissue section, okay, they do not really demonstrate those nice peplimers. Those are sort of obscured. The coronavirus particles are generally phagocytized by intestinal epithelial cells at the tips of the villus, and then within lysosomes, they form these viral factories. And generally, most of the coronaviruses that we see have a very dark nucleocapsid, but occasionally you can see some of them with these electron-lucent nucleocapsids. Some viruses are very oddly shaped, and they're very characteristic. And we see these particular virus particles that look like bullets. And these are rhabdoviruses. There are a number of types of rhabdoviruses. None cross-react with each other, okay? But very characteristic shapes. This is rabies virus, but it could just as well be vesicular stomatitis virus or lyssavirus of bats, etc. But very characteristic appearance for rhabdovirus, not one that you will mistake. And in tissue section, as you can see here, in the salivary gland of a skunk, you can see that in the affected epithelial cells, you have these large electron-dense inclusions of viral microfilaments and nucleocapsids. And eventually the viral particles will be assembled, the cell will die, and they will be released into salivary gland lumen, where they can mix with the saliva and be passed into the unsuspecting host. So that's something else you want to look for in cells, the presence or absence of viral inclusions. So location, presence or absence of paracrystalline arrays, presence or absence of inclusion material, especially cytoplasmic inclusions, and a susceptible host will usually give you all the information you need to come to an appropriate diagnosis. Here is another type of virus, a filovirus, with a very characteristic shape. This is called a shepherd's crook, and really all the filoviruses look like this. These are some very dangerous and deadly viruses, especially of non-human primates and human primates, such as Ebola virus and Marburg virus. These viruses affect the endothelium. They also affect the liver and the spleen and cause extensive damage. No other virus looks like this. So when you look at this, you can't tell whether it's Ebola or whether it's Marburg, one of the other filoviruses, but you certainly tell that it's a filovirus. In tissue section, these also will result in cytoplasmic viral inclusions. These are viral particles. We're looking at the nasal epithelium of a goat, and these particles are these very small electron-dense particles are retrovirus. This is an enzootic retrovirus which causes the formation of nasal tumors in epizootics in sheep and goats. This is a type D retrovirus. Well, I do like to superimpose a couple of things on a slide so you can use what you've already learned and put it into practice. We are looking at a section of several cells from a pig. I'll give you just a moment here. You can slow down the disc and look at the DVD. Okay, so I didn't tell you where this was from. Hopefully you identified the fact that we are looking at cross sections of several epithelial cells. The epithelial cells have a microvillar border of the same length, so that should tell you that we are looking at GI tract. This is intestine. Normal structures that we see include tight junctions and desmosomes here, and tight junctions here. We see more desmosomes along here. We have probably cross section of what appears to be 3 or 4 cells. One here, one here, one here, and one here. I see fat globules. I see mitochondria as normal structures. Let's go back. There is a nucleus. Here's some more mitochondria. I can see some rough endoplasmic reticulum, some interdigitating membranes, and then there are some very dilated profiles of what looks like endoplasmic reticulum. So we've described all the normal features we can, and then I would say that this is GI tract, possibly intestine. Next thing that we would describe would be all of the degenerative changes, and that you can see that the 2 cells in the center have marked changes that are associated with acute degeneration. We can start by the fact that the cells are swollen, and the cytoplasm is electron-lucent, so there is cytosolic swelling. There is loss of the surface substructures, which are actually the microvilli, although the tight junctions and desmosomes are intact. There is multifocal dilation of the endoplasmic reticulum, which contains numerous small electron-dense, non-enveloped viral particles. One of the cells has a large nucleus which appears to be swollen, is likely out of place because most of the nuclei in the intestine are basilar. Okay, we know that the cytoskeleton is damaged in this cell because of a loss of the microvilli. So this nucleus is probably free floating. There is marked chromatin lysis, and within the nucleus there are numerous small viral particles in paracrystalline array. Now at this point I want you to think, okay, can you have a virus particle that is in both the nucleus and the cytoplasm? It really doesn't work. So when you see viral particles in the nucleus and the cytoplasm, I want you to consider the possibility that we may have 2 different viruses. Okay, the ones that are in the dilated endoplasmic reticulum are not in paracrystalline array. The ones that are in the nucleus are in apparent paracrystalline array. So it's very likely we have 2 viruses, and that is exactly what's going on here. This is a young piglet. The piglet is affected with rotavirus, which is a RNA virus which forms within the dilated endoplasmic reticulum. in the cells at the tips of the villus, and it is co-infected with a common virus of young or immunosuppressed animals, and this is adenovirus. And it's adenovirus, a DNA virus which lives in the nucleus in paracrystalline array, co-infection. What a great picture this is. This would be an excellent picture for a examination. Just a little closer look at adenovirus. This is a chimp adenovirus in the liver, and you can see up in the top right the remnants of the nucleus, and there has been marked lysis of chromatin, okay, and we can no longer see the nuclear membrane. The virus particles are primarily hexagonal, and they are in paracrystalline array in the nuclear remnant. You can even see virus particles in the cytoplasm. I said you don't normally see that, but when you have almost total lysis and loss of the nuclear membrane, then right before that cell ruptures, you will have some viral particles in the cytoplasm. We're looking at the nucleus, close-up of a bladder epithelial cell from a dog, and within the nucleus we can see very small viral particles in paracrystalline array. This is another virus. This is morbillivirus. Generally with morbilliviruses you will see the virus particles within the nucleus in paracrystalline array, and you will see large cytoplasmic inclusions, which look very much like this. Okay? You can see right here. These aren't viral particles, but these are viral proteins. And the virus itself is within the nucleus. Canine morbillivirus. Another virus which looks pretty characteristic to me are herpes viruses. And we are looking at the lung of a cat, which is infected with herpes virus. And within the nucleus, as herpes virus is the DNA, you will see these viral particles which look like a small bullseye. They have a very dense nucleoid, and then the edge of the nuclear capsid is very dense, so it looks like a target. When the viral particle buds through the nuclear membrane, and we're stuck right here in the cisterna, as it buds through, it will pick up an envelope from the nuclear membrane and will resemble this particle or this other particle up here, still retaining that bullseye shaped appearance, which is characteristic for herpes viruses. Here is another herpes virus, once again budding through the nuclear membrane, picking up that envelope. And this is herpes B virus from a gibbon. So very characteristic looking bullseye-shaped virion, which is present in the nucleus of the cell. Just a nice picture of the human lymphotropic virus which causes HIV. And you can see the number of virus particles which are attaching to this macrophage. Excuse me, this is a lymphocyte. And this is also a case of lentivirus infection. This, take just a moment, this is a monkey. Hopefully you can tell me what organ you're in. Well, scattered throughout the section, there are large numbers of myelinated nerves, some of which are demyelinating. We have marked myelin splitting. There is demyelination. Probably outside the field, there are large numbers of macrophages cleaning this up. And you can see these very small viral particles throughout this area. And if you take a look at the inset, a characteristic feature of lentivirus particles is this triangular nucleoid. Realize that it is something that is somewhat fortuitous to see properly. You have to section them just so, otherwise they will look round like herpes viruses. But if you get one that's section appropriate, you will get a very nice triangular nucleoid. So those are lentiviruses, somewhat characteristic. Viral location, nucleus. Paracrystalline array, yes. One of the characteristics of this particular virus is this very large florid keratin cytoplasmic inclusion. And all of these wavy lines in the cytoplasm, this is all keratin. This is what happens with papillomavirus in the skin of dogs. Okay? DNA virus, paracrystalline array, and you get these lovely keratin inclusions. This is a coilocyte, a papillomaviral induced change where you have chromatin lysis. The nucleus itself is enlarged. It is glassy appearing, and if you look closely, you can, in a few coilocytes in any section, see viral inclusions. The prep, there's a very old preparation is very interesting, but we're looking at an epithelial cell from a squirrel. And to help you out, here is the nucleus here. There is a cytoplasmic invagination within the nucleus. That's actually cytoplasm here. Okay. Where are the viral particles? The viral particles in the cytoplasm. Are they in paracrystalline array? No, they are not. Is there an inclusion? Yes, there is a very large inclusion. And when you look at the shape of these viral particles at slightly higher magnification, you will see that the nucleoid here is dumbbell shaped. This is very characteristic for pox viral inclusions. We are looking at squirrel pox, which is caused by lepropox, and gives us lovely, bright, large, bright eosinophilic inclusions within epithelial cells. Just to finish up viruses, these are some of the things we've looked at before that people have confused for virus particles. Upper left-hand corner, we have nuclear pores. We have granules from mast cells, which can be confusing. People think that it looks like viral proteins, like we saw with morbillivirus. Here is a multivesicular body. And even glycogen clumps may be, this is actually a viral case, but even glycogen clumps may be mistaken when you are looking for a possible viral inclusion. It can be very tricky at high magnifications. Okay, so hopefully you've learned a little bit of something about how to approach describing and identifying viruses in electron micrographs. Let's move on to bacteria. A couple of general words about bacteria that, as we've seen in the slides earlier in the lecture, pretty much all bacteria look alike. Some have thinner cell walls. Gram-negative bacteria have somewhat thinner cell walls. Gram-positive bacteria have somewhat thicker cell walls. But for the most part, a bacterium is a bacterium is a bacterium. We'll look at some special types which can be identified on their morphology. But once again, we are often looking at the location of the bacteria, the susceptible host, and playing the vet game. Now, we are looking at the intestine of a rabbit. Okay, we have microvilli along these cells. These cells are largely degenerate. And attached to one of these cells, a cell which has a nucleus which is enlarged, It has chromatin lysis, a cell which has lost its microvilli and is bulging into lumen, so we can assume that we have degeneration and breakdown of cytoskeleton, and a cell that has obvious endoplasmic reticulum dilation. We can see that what used to be the microvillar surface is lined by these round to oblong structures which are densely osmophilic, don't really have any specific internal structure. There are some cleared areas in there, and this is what most bacteria look like on electron microscopy. You rarely could see any internal structure on bacteria. But these bacteria are doing something very specific to the cell. We know what the host is, and these bacteria are attaching and effacing E. coli. And a very characteristic cellular change that we see with E. coli that attach to the cell surface is the formation of what are known as cups. And here's a nice cup right here, and the bacteria is sitting right in that, or pedestals. So the cup and pedestal formation is very characteristic for attaching and effacing E. coli. And here we can see that the cell that they're attached to is degenerating. Cells next to it are either necrotic or also degenerate. Electron micrograph of numerous cross and tangential sections of bacteria. Can you tell what bacterium that is by looking at it? I can't. Okay, but this is a pig intestine. We can tell, maybe not intestine, but certainly GI tract by the fact that you have a prominent level microvillar border. So we're in the GI tract. Okay, the material that you see between the bacteria is the glycocalyx of the bacteria. And these bacteria are causing severe diarrhea without even touching that microvillar border. Those cells look excellent. Okay, this is enterotoxigenic E. coli. I've mentioned before, I will remind you that there are a number of bacteria in nature that want nothing more than to be a cilium. And these bacteria are generally found on or around cells with cilia, and we would have to think of a couple of possibilities. Mycoplasma is almost always found around ciliated cells, Bordetella, and Carbacillus. We are looking at the upper respiratory tract from a turkey, and you can see that there's a cross-section of several epithelial cells which contain both microvilli and cilia, these long cilia. If we could look a little closer, you could see the axoneme within with the characteristic 9+2 arrangement of the dynein subunits. Now, I want you not only to look at the presence of cilia and the presence of these bacteria which are enmeshed in it, so I'm going to think of those three different types, but remember we have to describe the normal features of these cells, the abnormal features. You can tell that while we might not have microvillar or ciliary loss here, we do have marked cytosol swelling and lucency. We have chromatin lysis and nuclear swelling. There are some hints of swelling of the endoplasmic reticulum, and these cells are bulging slightly into the lumen. So, evidence of acute cellular degeneration. And then finally, the presence of numerous cross and tangential sections of bacilli, which are enmeshed within the cilia. And this is Bordetella avium in the turkey. This is from a rabbit, and you can see that we are at very high magnification. We are looking at the cilia, and scattered within the cilia, oriented the same way, are these long filamentous bacilli. This is carbacillus. They have no internal substructure. You cannot see the microtubules within the bacteria that you do in the cilia. Okay, normally, if you're looking at a mouse or rat or rodent with carbacillus, you can put a silver stain on the tissue and that will color the bacteria, and you'll see a very dark stripes within the cilia lining the airways. And then this is what it looks like on electron microscope. We are looking at the inner ear of a pig infected with Mycoplasma hyarinus. And of course, in the inner ear, you also have ciliated epithelial cells. And here you can see enmeshed within the epithelium these bacteria. And they look different than bacteria that we've seen before because they are much lighter. To me, they sort of resemble degenerate mitochondria. And this is Mycoplasma. Mycoplasma is a special type of bacteria which has a very, very thin cell wall. Okay, this is why the normal antibiotics that you would take for a bacterial pneumonia do not work well against Mycoplasma. We have to take special antibiotics because antibiotics generally work on a cell wall. These organisms don't really have one to start with. So antibiotics that we use for other bacteria are useless against them. And they don't look much like bacteria. This is why I spent probably about 60 hours as a young first-year resident looking at the lungs of green turtles for bacteria, not recognizing these as bacteria. I thought they were probably mucus droplets or some sort of trash. I spent 60 hours looking at Mycoplasma, not even knowing that I was looking at it. I was looking for something that looked more like bacteria. We are in the stomach of a cat, and you can see numerous Helicobacter organisms, and these are helical bacilli. And at the bottom of these sometimes, or at the top, at one pole, you will see an axoneme. So that's cilium, so that 9+2 arrangements of microtubules and dynein, because these bacteria have a polar flagellum. And a flagellum is like the axoneme of a sperm, and it is this particular arrangement that is conserved throughout nature. This is Helicobacter acinonyx in the stomach of a cheetah, and gives you a slightly better look. This is sort of a more gracile looking bacillus with more twists in it, but you can see a very nice section through the axoneme of the polar flagellum. We actually looked at this picture earlier, and I'm just putting it in here to say that bacteria look like bacteria look like bacteria. This is a macrophage in a fathead minnow infected with Mycobacterium. And you can see the large number of bacteria within the cytoplasm of this macrophage. They show absolutely no ill effects because they have a very complex cell wall with wax D and mycolic acid, and it makes it very resistant to digestion. This is a case of yersiniosis in an African green monkey. And once again, very difficult to tell what the bacteria is. If I asked you what cell type this is in, hopefully you would see that the cell has very long filamentous pseudopodia, and you would tell me that this is a macrophage, and you would be correct. If you notice all of the little black granular material, you will see this occasionally on electron micrographs, especially infectious agents, and this is immunogold labeling. And immunogold labeling, you can label a particular bacterial agent, and that way you know on ultrastructural analysis that you're looking at that agent, that this cell is truly infected with Yersinia pestis and not another contaminant like E. coli or Pseudomonas, because of the very specific immunogold labels surrounding all of those bacilli. We're looking at the heart of a green sea turtle, which is for very high magnification. And we're looking at a particular type of bacillus that has a very characteristic appearance. And this is Chlamydophila or chlamydia. And chlamydia has several forms. The hardy, environmentally resistant form is the elementary body. And we're looking right here at elementary bodies. They look to me a little bit like norepinephrine granules. They have a very dense core. They have a membrane around them, and that core is generally eccentric. Okay, when these bacterial forms get into a susceptible host, they will mature into much larger forms which are able to synthesize RNA and DNA, and these are the reticulate bodies. And in between, there is a third form known as the intermediate body, which has some morphologic characteristics. Both, here's a very nice one here. You have that reticulate pattern of the reticulate body, and you still have part of that dense nucleus, which is the elementary body. Once again, we are looking at chlamydia. Here are the forms that we are used to seeing: the elementary body, the reticulate body. They're in large vesicles or vacuoles in the apical cytoplasm of this pig intestine. Here's a fairly uncommon bacillus. This is Neorickettsia rusticii, which causes Potomac horse fever. And early on, this was considered to have a quote-unquote trilaminate wall. And here's a picture, but it really does not. It does have a very thin cell wall, very difficult to see. But the outer membrane of that actually is the host phagosome. So this particular organism has the ability to get within phagosomes, turn off the ability of the phagosome to fuse with the lysosome, and this is how they live very happily within phagosomes. Hopefully you can see what cell you're in because of this irregular brush border. It's on tangential section, something that you need to get used to looking for. And this will tell you that you're in a proximal convolute tubule. But if you look closer, you can see that among these microvilli enmeshed here are these curvilinear bacteria, which are actually within the lumen of the proximal convolute tubule. These are leptospirers. This is leptospirosis in the pig. So these are curvilinear bacteria, and there are a couple of bacteria which are curved. Leptospirers generally live outside of cells, and within cells, Lawsonia live in the apical cytoplasm, another characteristic curved bacteria. Once again, here is our friend Yersinia pestis with the immunogold labeling, and you can see that the cell wall is delineated here. It's very electron-lucent, it's not very thick. So this is the cell wall. This is the interior of the bacteria, and they just don't have much structure. Okay, this is your prototypical gram-negative bacteria. Gram-positive bacteria generally have a much thicker cell wall right here. Okay, so it's very thick. And this is Bacillus anthracis. Bacillus anthracis is a spore-forming bacteria. We are looking at the spore right here. But thick cell wall, much thinner cell wall. Okay, very difficult to rely on that, but that's what it's supposed to be. Okay, moving on to protozoans. I want to start with the apicomplexans. And much like bacteria and viruses, there are a number of apicomplexan species that we're familiar with, but they all essentially look alike. And we often find ourselves relying on the host and the cell that they invade to come up with an appropriate differential diagnosis or diagnosis. And there are some very characteristic features that you will see in apicomplexans, most of which go toward initial invasion and formation of the parasitophorous vacuole within the host cell in which the apicomplexan resides. Things that you will see which will identify apicomplexans is the conoid ring at the top. Then these cells have what are known as, I'm going to show you on the cartoon first, small micronemes and larger rhoptries, which are much larger. Okay, here's your conoid ring, here are your smaller micronemes and your large rhoptries. And all of these essentially are used within the first 20 minutes of invasion to get the parasite within the cell and to form an appropriate safe habitat for it. These merons also generally have a terminal nucleus, and they often have granules, and some people call them amylopectin granules or starch granules. They often have granules within the cytoplasm. Let's look at a prototypical apicomplexan. And normally when you are looking at apicomplexans on electron microscopy, you'll see 8 or 4, or even more within a single cell. Because we're getting a very thin slice, it is very unlikely that you will get all of the features. But if you look at several, you will often see the features that you need to make up the diagnosis. This is a rare occasion. This is a apicomplexan that has pretty much everything in the same field. Here is your conoid. We have the very small micronemes. We have the much larger rhoptries. Here are your starch granules throughout here, more micronemes. Larger ones are rhoptries. Okay, here's your basal nucleus. They also have mitochondria, they have Golgi bodies as well, but it is unlikely that you're going to see that in all of them. We are looking at a Brady cyst. Okay, so this is a cyst within the heart muscle of an opossum. This is toxoplasma. And we can identify a lot of these structures, but rarely can you identify everything. Here is one one zoite, where we can see the conoid, we can see the micronemes, we can even see a rhoptry, but I don't see a nucleus or many starch granules. Here's one where we can see part of the nucleus, and we can see the micronemes, and this may be a rhoptry, but the starch granules are much more apparent in these over here. This one we can't see the conoid. So you often have to look at several of the structures. This is a Brady cyst. There is a cyst wall around it, separating it from the muscle fibers. Just close up, really nice picture of a toxoplasma schizont, which is invading an intestinal epithelial cell, and a good look at the conoid ring and the microfilaments, which assist. The conoid ring is the first thing to puncture the cell, and then the microfilaments contract, drawing the organism through that conoid ring and extruding it into the parasitized cell. We are looking at the intestine of a cat, and once again we see cross and tangential sections of numerous apicomplexan parasites. This is the only ring one, it appears, on this edge where we can see a conoid ring. Okay, we have rhoptries here, we have some micronemes. So you have to sort of look at several of them. One thing that we see very nicely here are the amylopectin granules and the nucleus. But the other structures are somewhat difficult to see. Something else that you can say is that there is no cyst wall here. These zooites are living free in the cytoplasm of this parasitized cell, and this is characteristic for Sarcocystis. We're looking at the intestine of a cat. See the microvillar border here, all the same size. But we're looking at the gametes. The gametes this is the microgamete, and this is the macrogamete. And all of these large granules within the macrogamete, okay, essentially will form the hard wall when this transforms into the oocyst after fertilization. When you look at apicomplexans, you quickly realize that they all look alike. There's nothing that really sets them apart. Okay, this is Hamondia, but if you told me this is Sarcocystis, I really couldn't tell the difference. This is Neospora. Neospora has a very large cyst wall. This is surrounded by macrophages. And we can see all the features somewhere in one of these parasites that we need to see. Conoid ring is down here. Okay, we can see some rhoptries and micronemes, and is surrounded by a very thick cyst wall. This is the spinal cord of a dog. We have macrophages, we have numerous unmyelinated nerves, and we have this nice cyst. Another form of apicomplexan is Cryptosporidia. And Cryptosporidia is an interesting apicomplexan. Okay, the mature forms are called trophozoites. They contain zooites, and these are extracytoplasmic intracellular parasites, meaning that they sit on top of the cell. You can see the microvilli here. They sit on top of the cell. They attach with this feeder organelle, and there is a very thin rim of cytoplasm surrounding this trophozoite. Here are your zooites within. And so they essentially live inside the cell, but outside the cytoplasm. All we're looking at right here is a very thin rim of cell membrane. I want to go back for just a moment to this other. This is a great electron micrograph of Cryptosporidia in the intestine of a hamster. Hopefully you also noted that there is significant acute cellular degeneration of these intestinal cells, which are bulging. There's cytosolic lucency. The nuclei are riding way too high. There's chromatin lysis. But also within the apical cytoplasm, you see these aggregates of curvilinear bacteria. This is what we call in the trade a twofer, or a double whammy. This is an electron micrograph that has not just one, but two infectious agents. So in a testing environment, you would be expected not only to identify the organ proper by knowing this is Cryptosporidia, you would be required to describe the degeneration seen in these cells, and to describe and identify the parasites Cryptosporidium and Lawsonia intracellulare. So let us finish this. I think this will be the last hour. I know that y'all are as excited about that as I am. We're still looking at apicomplexan parasites, and this is a very nice electron micrograph from an ox, maybe an ox who lives in Africa. And what we are looking at here is a blood vessel within, and I know some of you already figured that out, we're looking at the brain. Okay, it could be spinal cord, but we're looking at the brain because we have some myelinated nerves, and obviously you're going to have a lot of myelinated nerves in the brain. And we're looking at a blood vessel, and within these marginated erythrocytes, that's why we didn't recognize them at first, but they're marginated, they're sticking to the endothelium. Within them are generally two cross section. Some of them only have one, but again, that's a sectioning issue of apicomplexan parasites. And if you look closely, you might even be able to make out some of the structures that you would expect. And this is a condition called, well, the organism is Babesia bovis. This is a form of babesiosis that is seen, which is known as cerebral babesiosis or pink brain. And when you look at the brains of these animals immediately after death, the brains actually look pink because all of the capillaries are dilated and are filled with these attached red blood cells, causing sludging. You can see some small infarcts there too. But the presence of the Babesia causes these infected red blood cells to upregulate their adhesion molecules and stick to the walls of vessels. Probably this allows the parasite to survive longer in the red blood cells because they're stuck here. They're not going to circulate through the body, be identified by the reticuloendothelial system, and removed in the spleen. So it's a nice evolutionary change induced by the parasite and makes for a really nice story, don't you think? We're looking at the skin of a horse. This is a very complex image here. What I'd like you to notice is, as I said before, you will rarely get a electron micrograph where you can diagnose a true itis. It's usually 1 or 2 or 3 cells. But here we have 10, 15 or more cells, including several neutrophils. Okay, here, and here. We have a lymphocyte, and then we have 2 large macrophages. And there's some other cells. Here's another neutrophil, maybe more lymphocytes. But we have 2 large macrophages, and the macrophages have phagocytosed large numbers of leishmanial amastigote. There's cutaneous leishmaniasis in the horse, and we are looking at macrophages. And we can see that these amastigotes have some very characteristic structures. They all have a nucleus, and then they have these linear bodies here. Okay? And these are known as kinetoplasts. And kinetoplasts are essentially the mitochondria for the leishmanial amastigote. They do contain DNA. Okay? And very difficult to see on EM. It's often a sectioning problem, but here's a very nice one where the axoneme is pointing at the, on this side, it's pointing at the nucleus, and that is what we would expect. You can also see that these amastigotes have an axoneme. Okay, at one point they have a flagella. Okay, we don't know that much about how they use their flagella because, to my knowledge, Leishmania mastigotes, unlike Trypanosoma, a related organism, have never been identified in the extracellular environment. Normally when you see them, they have already been gobbled up by macrophages. Here is that Leishmania. We are looking at it in a blood vessel. Here's one that is pushing along a red blood cell, and you can see that they all have these paired axonemes or flagella. This is the tripomastigote form of Trypanosoma braziliense in the ox. Another protozoan that you're probably quite familiar with, but not very familiar with ultrastructurally, is Giardia. Giardia, we all know, has a number of flagella, up to 8, and you can count the axonemes here. I can only see 1, 2, 3 here. Here's another organism with a couple. Here's another one. Okay, that characteristic structure. Notice that as opposed to Cryptosporidia, the sucker plate, that's what it's called, it's a little different, of Giardia, does not do any damage to the microvilli which it attaches to. Okay, so it's a sucker plate, multiple flagella, does mild or no damage to the cell underneath, and this is Giardia. Another protozoan is Entamoeba. This is Entamoeba invadens, and we can see some things that are fairly characteristic of amoeba here. And amoeba have these lamellar stacked mitochondria, which are very characteristic here and here, very characteristic of the parasitic amoeba. Okay, we have a large central nucleus, and when you look at the karyosome or the endosome of, which is essentially within the nucleus of the amoeba, it looks like a very large nucleolus, and this is what it looks like ultrastructurally. It's a little more disappointing. And this is Entamoeba, and you can see within phagolysosomes in the cytoplasm, you can see that it has been gobbling up bacteria, which it feeds on. We are in the lung of a macaque, okay, and this is the alveolar wall. Here is one capillary, which is full of blood cells. Here's another. Looks like we have a endothelial cell here. We have inflammatory cells, which are marginated within the wall. And you can see that the wall is covered by type 2 pneumocytes. So there's marked inflammation. Okay, these all look to be either histiocytes, possibly a lymphocyte in here. Okay, maybe another histiocyte here and some debris. Okay, and then these type 2 pneumocytes. So there's been damage to the wall. You can see this type 2 pneumocyte has some surfactant right here. Damage to the wall. We've lost the type 1 pneumocytes. The type 2 pneumocytes have taken over. This wall obviously is not passing any oxygen, because oxygen is just not going to diffuse through these cells. And within the lumen, you can see the cause of this. Okay, there are numerous cystic forms of a parasite known as Pneumocystis carinii. And Pneumocystis carinii, a number of them have been renamed. I don't believe that it's been renamed in macaques. It's still Pneumocystis carinii. As we develop more and more sophisticated technologies, we are subclassifying more and more, and they all used to be Pneumocystis carinii, whether you were dealing with a mouse or a rat or a dog or a monkey or a horse or whatever. Now they've been broken out, so we have Pneumocystis equinus, we have Pneumocystis muris, and I guess soon we'll probably have a lot of other ones, but this is still Pneumocystis carinii. And here we are looking at a macrophage, which is gobbling up, you can see the pseudopods, macrophage which is gobbling up a lot of these collapsed cyst forms, which we've looked at. And then here's one that you don't often see with Pneumocystis, and this is an intact trophozoite with the immature forms within. Most of what we see in the alveolus are collapsed dead cysts and cyst walls, or the trophozoite once the mature protozoans have been released. And usually what we see that causes that foamy cytoplasm, that sort of eosinophilic foam in animals on light microscopy which have pneumocystis, is a combination of these macrophages, these collapsed cyst walls, and surfactant, which is produced by those type 2 pneumocytes. Maybe even a little of edema fluid. But I like this picture because this looks very much like that eosinophilic foamy material, bubbly foam that we see in alveoli on light microscopy. So this is Pneumocystis carinii. Leaving the protozoans, we want to look at one similar organism, but this is an algae. This is Prototheca Zopfii in the skin of a dog. It's very difficult to pick up that you're in the skin, but we are looking at several macrophages which have ingested a number of the protozoal organisms here and here. Okay, they are collapsed, and one of the features that we can see both with light microscopy as well as the on EM is the endosporulating alga right here, and you can see it is forming that very characteristic tripartite shape of the endosporulating form. Some people look at that as a peace sign, some people look at it as a Mercedes-Benz emblem, but it's just tripartite endosporulation. Well, I would like to go ahead and finish this hour and this lecture by quickly reviewing a number of neoplasms. And as I've done with the infectious agents, I'd like to give you a couple of general ground rules when looking at neoplasms. All neoplasms, as we know, try to recapitulate the tissue that they are derived from. And we're looking at 2 or 3 or 4 cells within a neoplasm. So if you can identify tissue, if you have reviewed and gone back and become adequate in identifying a tissue based on changes in the EM, chances are you'll be able to identify neoplasm from that tissue because they usually share the same features. If a neoplasm is so poorly differentiated that you don't have the features that are seen in normal tissue from that organ, you probably will not be able to identify it. One other thing that I need to reinforce is that when you're looking at 2 or 3 cells, you really cannot assess benign versus malignant. That is much better performed on light microscopy. So let's move forward. Hopefully you'll take a little time, shut down or freeze the DVD. I don't mind, you can freeze it in mid-word and try and figure out where these come from. This first slide is from a neoplasm, I'll tell you that, but it could just be normal tissue. And hopefully you have recognized by the tubulo-vesicular cristae in the mitochondria and the numerous profiles of smooth endoplasmic reticulum that we are looking at a cell from a tumor arising in a sterol producing organ. This happens to be from an adrenal tumor. Is it a carcinoma or a adenoma? I can't tell you. And even on light microscopy, endocrine tumors are very difficult because they don't often have the characteristic features of malignancy. Even malignant endocrine tumors look very benign on light microscopy, and we end up looking for other features of malignancy. You'll very rarely see mitotic figures, almost never see atypical mitotic figures. Evidence of necrosis is something that the humans use for evidence of malignancy in endocrine tumors, and it seems to work pretty well in animal species. We are looking at a tumor also from the adrenal gland. This is from the medulla. This is a pheochromocytoma, and the cytoplasm is replete with dense core granules which are centrally located. So this is an epinephrine producing pheochromocytoma. If I didn't tell you this was from a tumor, you wouldn't be able to tell it from normal tissue. And here's one that's producing norepinephrine. I am hopeful that when I flash this picture up, you will also think, however briefly, that you might be looking at the elementary bodies from chlamydia. Because to my eye, this could be chlamydia. Unfortunately, these are free in the cytoplasm as well as it could be norepinephrine granules. Tissue from a ferret. Also, these cells are full of granules, but you can see that the nucleoid within these dense core granules is linear. And so this is a beta cell tumor or insulinoma. in a ferret. This is an ox. Well, we're looking at more cells with dense core granules. At this point, all you could tell me is, well, it could be a pheochromocytoma, it could be a neuroendocrine carcinoma, it could be, this turned out to be from a case of medullary thyroid carcinoma where they were demonstrating these dense core granules. But at this point, neuroendocrine tissue is about all that I could say. This is from the thyroid of a dog. And you can see that the field is composed of numerous cells with large central nuclei in which the cytoplasm is full of mitochondria. Going back to what we discussed when we talked about the thyroid gland, there are oncocytic cells, also known as Hürthle cells, in the thyroid gland, and this would be an oncocytoma. We're looking at several cells with sort of indented nuclei which have large numbers of filamentous cytoplasmic projection and then sort of wrap around each other. And there are a couple of neoplasms which have this morphology. Meningiomas and schwannomas, or tumors of nerve sheath origin, both do this. The difference between these two, and it can be very difficult, is that the meningiomas often have desmosomes which hold the adjacent cells together. And this is only seen in meningiomas. We don't see these in schwannomas. But you can see all of these filamentous projections. The cells sort of wrap around each other. Here is a neoplasm from the cervix of a rat. And the neoplastic cells, look, they have lots of euchromatin. They have a very large nucleolus. This cell seems metabolically active. Maybe it's a rapidly dividing cell. In the extracellular space, there's a large amount of filamentous collagen. But what I want you to see is that the cytoplasm of this cell is filled with large lysosomes which contain debris. And these cells have bright pink granules. These have been known as granular cell tumors. And one of the characteristics of granular cell tumors is the presence of these large lysosomes, which give it its characteristic pink, brightly eosinophilic character. This is a granular cell tumor from the cervix of a rat. Here is another granular cell tumor. And it also has these lysosomes, not as big and as prominent as the last one, but these are lysosomes as well. And this is a similar tumor, but this is from the skin of a rat. This is a granular basal cell tumor. It's a basal cell tumor with the granular variant also being lysosomes, also being brightly eosinophilic. So when I see tissues that are brightly eosinophilic, that the cells can be called granular. This would be the ultrastructure that I would expect with a granular cell tumor. We are looking at the skin of an owl. And these are, if you look closely at the inset, you'll see that very intricate scroll work that's associated with a number of the granules, here's some up here, of mast cell tumors. So this is mast cell tumor in an owl. And you remember, hopefully, when these degranulate, you'll get these empty holes where the granules once were. It's from the skin of a dog, and we're looking at cross-sections of several polygonal neoplastic cells whose cytoplasm contains abundant, clear, non-membrane-bound vacuoles, which are consistent with fat. This was actually from a liposarcoma, but I defy you to make that diagnosis based on this electron micrograph. Most lipomas, you would see a single large vacuole of fat, and here we see multiple small vacuoles, but there's no nuclear atypia, there are no mitotic figures. So once again, we're looking at a tumor of fatty origin or of fatty tissue, but that's as far as you can really go. We are looking at the skin of a dog, and we can see that there are cross-sections of several polygonal cells with abundant euchromatin, prominent nuclei, which have tight junctions and abundant keratin tonofilaments. And you can especially see those tonofilaments in some of those junctions between the cells. And this is a tumor of squamous origin. Okay, this is a squamous cell carcinoma. Once again, tough to tell benign from malignant based on a couple of cells. We're looking at a infiltrative neoplasm from the oral cavity of a dog. Very high magnification on one of the cells. And what I want you to take a look at are these elliptical structures, which have this sort of cross-hatching, and then there are some which are larger and darker. And this is what you see in melanoma or amelanotic melanoma. And amelanotic melanomas, you will have these pre-melanosomes, but very little pigment on them. So those are what pre-melanosomes look like in melanoma. What type of tumor is this? Well, just looking at the cytoplasm, especially up here, these are actually several different mast cell neoplasms, and the intricate scroll work should make you think about mast cells. And you can also see the same thing in basophils as well. We are looking at a spindle cell from the leg of a mouse with a large neoplasm, and you can see that this cell has very regular myofilaments. Here are the Z bands, here's the A band, if you want to call that, or the actin and myosin filament. Okay, it looks a little bit like that smooth muscle cell, but obviously a smooth muscle cell would not have these filaments in traditional array. Those only have those dense bodies. So this is a rhabdomyosarcoma. Can't tell benign from malignant here. Okay, rhabdomyomas would be extremely uncommon. And this is about as good as it gets. This is one of those that you probably didn't even need to run EM on. I would imagine that with a Mason's trichrome, these would stand out fairly nicely. This is what you tend to see more often. Okay, and you really have to look hard and you have to use your imagination. Okay, or something like this. You can see Z bands, you can see some myofilaments, abundant, robust mitochondria, but this is what you often see in rhabdomyosarcomas, ultrastructurally, very poorly formed myofilaments. That cell is trying to make myofilaments, it just does a very poor job. And we are looking at a cell out of a neoplasm in the GI tract of a dog. Okay, this very characteristic crimped nucleus, a spindle cell surrounded by collagen. But look at what we have here. These are those dense bodies. And this is a cell in a smooth muscle tumor. Whether it's a leiomyoma or a leiomyosarcoma, your guess is as good as mine. And our final image for this lecture is from the skin of a dog. And these are round cells. These are lymphocytes. They have almost no organelles to speak of. But the nuclei are very intricate. They are bizarre. They are what is known as cerebriform. This is very characteristic of T-cell epitheliotropic lymphoma in the dog. These cells will infiltrate the epithelium and have a very characteristic ultrastructural appearance. There are a number of excellent textbooks. These are somewhat older. There are newer editions out. Weider's Functional Histology is now in its 5th or 6th edition. And you also get with that a CD. I lifted a number of the cartoon images, the histologic images from this, and the color images from this presentation from that textbook. I think it's excellent. It has a lot of very good EM images in it as well, and even though it's primarily for humans to get a good grasp on basic ultrastructural anatomy and a variety of organs, I don't think you can beat that. Cheville's Cellular Pathology and Ultrastructural Pathology, there are newer editions available. Cheville's book on ultrastructural pathology in the 2nd edition is an excellent book, updated in the last several years, and you can't beat it in terms of learning basic ultrastructure and the infectious organisms. And finally, Gaudiali's Ultrastructure of the Cell and Matrix, a good book and exhaustive. Basically, it's human ultrastructure. It's a 2-volume set and has literally chapters and chapters on mitochondria, all the various organelles in the natural and disease state, and has excellent illustrations. With that, I will thank you for your time and attention, and I hope that you are able to glean some good basic information about ultrastructural analysis. As I promised, I have not made you an electron microscopy expert, but I hope that I have alleviated some of your tension and fear about approaching electron micrographs. Thank you.